Well, good morning, everyone. So thankful to have all of you here today in person, as well as everyone that's here virtually logged in. This is a really special day. It's our second annual symposium. Last year, we gathered at PCOM, and this year, we're very, very thankful to be welcomed by OU. Thank you all so much. At this point, I would like to actually turn it over to one of our very special and important sponsors, Cheryl Graffinino from the Osteopathic Heritage Foundation, just to say a few words.
Good morning, everyone. It is such a pleasure to be here with all of you. And for those of you I haven't met yet, I am Cheryl Graffinino, and I'm a program officer with the Osteopathic Heritage Foundation. It's my pleasure to welcome you to the second annual Alzheimer's Pathobiome Initiative and PCOM Symposium. And on behalf of the Foundation's Board of Directors and staff, thank you to Dr. Brian Balin, Dr. Graciela Muniz-Torreira, and Nikki Schultek for your leadership in bringing this important event to the Dublin campus of the Ohio University Heritage College of Osteopathic Medicine. The Foundation's heritage began in 1940 with the founding of Doctors Hospital in Columbus, Ohio, by three visionary osteopathic physicians, Dr. James Watson, Dr. Harold Clybourne, and Dr. Ralph Licklider. Their commitment to accessible, high-quality, holistic healthcare laid the groundwork for what would become a high-quality regional health system and one of the most respected osteopathic training programs in the United States. In 1998, the asset sale of the Doctors Hospital system to Ohio Health provided an influx of funds into the foundation to advance our mission to improve the health and quality of life in the community through education, research, and service consistent with our osteopathic heritage. Since then, the foundation has approved more than $338 million in funding, including more than $266 million of support for osteopathic medical education and research locally, statewide, and nationally. This work is only possible because of the dedicated board of directors of the foundation, and I want to recognize those that are here today. Dr. Rebecca DeVillers, Dr. George Fairber, who I think will be joining us shortly, Dr. Robert Palma, Mr. Rick Vincent, and Ms. Terry Donlin-Huseman, the Foundation's president and CEO. Thank you for your leadership and commitment to our mission. The impact of the Foundation's investments can be seen on the campus that we're on today. This campus was made possible through the Foundation's transformational support of Ohio University's Heritage College of Osteopathic Medicine. With more than $193 million in support approved, the Foundation is proud of the accomplishments of the Heritage College, including its designation as the largest public medical school in Ohio, the leading osteopathic medical school in the nation for NIH funding, and its contributions to Ohio University earning the Carnegie R1 designation. The foundation has also invested more than $22 million in endowments to support biomedical and translational research at colleges of osteopathic medicine across the nation, including support for Dr. Balin and Dr. Muniz-Torreira. Dr. Balin holds the Osteopathic Heritage Foundation Endowed Chair for Aging Research in honor of Ruth E. Purdy, D.O., George O. Fairber, D.O., who is among the directors who will be joining us today, and Peter E. Johnston, D.O. Dr. Muniz-Torreira is the Osteopathic Heritage Foundation Ralph S. Licklider, D.O., Endowed Professor in Health and Aging. As I shared with some of you last night, the collaboration between Brian and Graciela began just a year and a half ago at a convening of the Foundation's endowed researchers here in Dublin. We're so very excited about their collaborative success since then and look forward to seeing what they accomplish next and what is accomplished as a result of all of you coming together here in Dublin today. Again, we're honored to support this pioneering work to better understand the role of microbes in Alzheimer's and other dementias, and I look forward to a day of shared learning and discovery with all of you. Before I end, I would very much like to acknowledge Brian Thompson and Dr. Dean Burke, who is not with us this morning, but they are instrumental in events like this happening on this campus. And so if you see them today, please thank them for the experience that you've had. So now I'll turn it back over to Nikki to begin the day.
Cheryl, thank you. So wonderful. We're so grateful. So a little bit of general business. In front of you for our people that are here in person, you have a speaker button. just to be aware of. So if when the opportunity arises for Q&A, so we ask that after each presenter, we're not going to do questions, but there will be specific times just before the break and lunch, as well as during the panel discussion later, where questions for the speakers can come up. Also, we'll have our teammates monitoring the virtual chat. So for those of you tuning in online, please go ahead and put your questions there, and we'll go ahead and respond throughout the day. Thank you so much. What you'll notice about this year's event is that it's broader. So last year, our Alzheimer's pathobiome team came together at PCOM around the concept of bridging silos to better understand how infections and the host response in the microbiome can get involved in Alzheimer's disease specifically. This year, we're actually wrapping our arms around diseases that impact the brain and have to do with the pathobiome, which is, I like to say, the group of organisms that are kind of being jerks. They may be collaborating together to cause illness or disease. We're also going to be looking at the microbiome and the host response, the immune response, but we're looking at it across the human lifespan because we know that these infections can cause trouble literally from toddlers through advanced age. So I am actually very privileged to say that as of December 2024, I founded a center at PCOM called the Pathobiome Research Center. Prior to that, I've had the privilege to collaborate with Brian Balin and many of our other teammates for about 8 years. Building collaborations, I call it collaboration architecture, getting people together that might not normally have the opportunity to collaborate in an interdisciplinary fashion to really move forward these concepts and to avoid mistakes and make sure that we are making the most of precious patient samples. And the thing that I think I want to underscore about myself is that I am an IACI patient. I'm going to use that word in my presentation, and I'm going to introduce you to it. Infection-associated chronic illness. And that is a sort of larger catch-all that encompasses a whole variety of conditions that can develop from things like Lyme disease, which is a bacterial infection. And with this, I want to express that the whole reason we're here today is for patients. So I want to begin the day with that intention, that all of the researchers that you will hear present are singularly focused on reducing suffering. People that right now might be tuned in virtually that are not well enough to be here. I know you're there listening, interact with you. You are seen, you are heard. We are also here for people that lost people that they love to these diseases. We are here for Alex. We are here for Russ. That's what we're here for. So we begin the day with that. Last year, we were so thrilled. Science Magazine featured our event that took place at PCOM, generating sort of an unprecedented level of exposure to this whole idea, right? And if you had told me 6 years ago that that would happen, I wouldn't believe you. So we're so thankful this year to be looking at this broader issue. So this slide really covers a lot of ground. And I'm happy to get anyone a 5-page list of references that would like them. I'm going to reference a lot of work. There were too many to put them on the bottom in those little tiny footnotes. So across the human lifespan, we are acutely aware that infections can get involved with the immune system and host genetic factors and other elements of things that we are exposed to throughout our lives to lead to a disease process. And in this case, I'm really focused on the brain, right? So PANS and PANDAS, you'll see here, the P in those conditions stands for pediatric. These are pediatric autoimmune neuropsychiatric disorders, can be associated with streptococcus, strep throat, or other infectious agents like mycoplasma, Borrelia burgdorferi, which is the bacteria that causes Lyme disease, and others, even viral infections. Then you have people that are in their adolescence and teenage years that may go on to develop schizophrenia. Schizophrenia has been associated with Toxoplasma gondii, which is a parasite, also with other infectious agents such as Bartonella henselae, which is also another vector-borne pathogen transmitted by ticks, fleas, and otherwise. Then you have multiple sclerosis that's been strongly associated with Epstein-Barr virus, so much so that in work that came out of Harvard by looking at health records data, the researchers concluded you could nearly say that if someone hadn't been exposed to EBV, they did not go on to develop multiple sclerosis. Now, what we don't understand is how these individual infectious agents are behaving. We don't know if some of them are gatekeepers to other infections, such as the case with HIV, where the immune system is affected and then other things come on board. We don't know if certain other things precede this, like issues with the brain's protective shield, the blood-brain barrier, familial or genetic issues, things that you're exposed to, that make you more vulnerable. Then you have Parkinson's, which has, a body of work emerging on the potential role of the gut microbiota and other infectious agents. And then last off with Alzheimer's, across this spectrum of age and human lifespan, Alzheimer's now at this point in time, this past week having discussions, with our team and a number of other individuals, we agreed that the concept that microbial infection could get involved in Alzheimer's is no longer something that is a fringe idea. It is accepted and has come to a point where, yes, we would like to understand it better. And that's why we're here today, right? We need to figure out exactly how mechanistically infections can, in certain individuals, trigger neuroinflammation. How can they trigger, issues with autoimmunity, right? Such as that are seen in PANS and PANDAS. And today we have some of the world's experts in these areas, including, PANS and PANDAS, one of the biomarker panels, the Cunningham panel. Dr. Madeline Cunningham is going to give a talk today. That panel is a life-saving panel because it demonstrates that a child that has this neuropsychiatric syndrome actually has autoantibodies against basal ganglia. And this is incredibly important because it permits insurance coverage for life-saving IVIG therapy. So the whole reason I'm standing here, and I'm so lucky to be standing here and feeling well, is that, as I shared, I'm an IACI patient myself. It started 10 years ago with symptomatology that was body-wide, and it literally affected nearly every system of my body. But the most disturbing symptoms were the ones that affected my brain. I had neuropsychiatric symptoms, cognitive symptoms, and symptoms that looked like MS. I do not have multiple sclerosis. I've had multiple brain MRIs. I do not have lesions. But what my doctor told me at the time was truly disturbing. My neurologist said some people just sort of simmer like you for a decade, and then eventually they develop lesions. So I can't tell you today that you don't have MS. And this was crushing. You can see in the picture my son Owen there with me in bed. I had sunglasses on in bed because I was having this terrible stabbing eye pain. And my kids were 3 and 5. So my future was uncertain. But what I will tell you is the great news for me is that I had the ability to read medical literature. I worked in pharmaceuticals and biotech. And after I had 12 specialists that were focusing on these different systems of the body, but not why a healthy person who is running half marathons would suddenly be so ill, I ended up finding Dr. Charles W. Stratton's work. Dr. Stratton passed away almost 4 years ago, but he's with me here every day as I work forward. So he had published research linking the chlamydia pneumoniae organism that you'll hear Dr. Balin talk about a little later to some of the disorders I had been diagnosed with, including interstitial cystitis, the bladder pain disorder, as well as a potential driver in multiple sclerosis. Research had demonstrated that the C. pneumoniae organism could be getting involved there too. So I reached out to Dr. Stratton, and as I say, the rest is history. He became my physician. We had testing. It turns out that I was living in Connecticut. I was infected with tick-borne pathogens. I had this very strange air hunger and rib pain. I only later learned was Babesia microti infection, a parasite infecting my red blood cells. I did have lifelong asthma too, and that facet of my work is really interesting also. Asthma, 50% of it can be placed into remission with macrolide antibiotic therapy. And so I'm very pleased to say that as a part of a team of individuals, an incredible team, we have something called the I Treat PC clinical trial, which is funded by PCORI, where we will have 3,200 patients prospectively in RCT. And one of our arms is azithromycin for asthma. So we're going to put some serious numbers behind this situation. Also screen for 2 organisms that I had likely been infected with my whole life, driving severe asthma since my childhood, chlamydia pneumoniae and mycoplasma pneumoniae. But being well today is a gift. I feel like I was given a second lease on life after being so sick. And that's why I stand here with all of you. And this is why I thought, all of this happening to me has also led to people around the world contacting me and saying, Nikki, have you thought about and has your team thought about these conditions? So I'm going to bring PANS and PANDAS to the forefront here because you just saw what my symptomatology was as an adult with one of these chronic infectious processes and with immune activation and immune dysfunction. What does it look like in children? And I'm going to read just a staggering quote. 10 adolescents picked at random with mental illness severe enough to require hospitalization. 9 out of 10 had evidence of tick-borne infections. 9 out of 10 had evidence of autoimmune encephalitis. We're talking about a brain on fire here. We're talking about causality, right? And understanding, not just characterizing a patient and saying, oh, you have psychosis, you have schizophrenia, you have OCD, a severe eating disorder, but really understanding something that can potentially drive it. And in these children, the underpinnings of the pediatric autoimmune neuropsychiatric conditions associated with various infections have similar biological underpinnings to the adult conditions that we study. Neuroinflammation, issues with autoimmunity, immune dysfunction, issues with the blood-brain barrier. So you start to see these common threads. And so we really feel it's important, that we hear the patients and we look at the literature across a broad range of life stages. And so we're doing that at the Alzheimer's Pathobiome Initiative. Even though we're called AlzPI, we're looking at other brain diseases because we know that we can learn from other disciplines and also avoid mistakes and spur innovation. And this one made me laugh and I had to share it. So with everything that I'm saying, right, if we have evidence suggesting that infectious agents can actually be driving disease in some patients, why the heck does it feel like it's the elephant in the middle of the room that nobody's looking at? So here's the elephant here feeling kind of upset about that. But there are some very powerful reasons for this. Some of them come down to human behavior and dogma, right? What we presume to be fact, just the whole concept of acute infections being acknowledged, but our acknowledgement of chronic infections is more limited, how they can contribute to long-lasting sequelae. We know about HIV. We know about HIV neurocognitive issues, hand, but we know syphilis can cause dementia. We know Borrelia burgdorferi can produce a Lyme dementia, but yet broadly, patients aren't being screened. One of the other powerful reasons that this is the case is economics. So in terms of drug development, antimicrobial drugs are some of the least profitable in the world. And this is a major problem because it means that the potential solution to some of these problems are just not profitable. And that can create very big challenges. However, being a glass half full person, I point out the fact that there is ample space in the diagnostics realm for advancement around these ideas to bring very profitable things to market that also spur the economics in other realms. We also have the possibility of therapeutics maybe that aren't traditional antimicrobials. What if we had individuals that we could understand why their body might harbor Borrelia burgdorferi, for example, but they're not clinically ill? We can understand if there's some sort of tolerability piece that could be exploited in terms of drug development. So speaking of our interdisciplinary collaboration, and thanks to AI, I was able to cook up the image on the bottom, which would be my dream. Not necessarily having the brain floating in the room, that could be creepy. But if we could all be together as advocates with knowledgeable patients, clinicians, researchers from different disciplines around the table, in an institute, this would be amazing. That's my pie in the sky or my brain in the sky dream. But the reality of it is on top. This is how we collaborate, is mostly on Zoom. Our team convenes once a week for many years now from all different disciplines, talking about all the various ways that our consortium is moving this forward, everything from basic research to harnessing the literature and publishing to things like our meeting today. And I'm so grateful for my AlzPI teammates that are here. We have been harnessing knowledge that's already in the literature, and we feel this is incredibly important. This year, we published this very important piece with Dr. Praveen Bethini as our lead author, with the guidance of Dr. Lavinia Alberi, who couldn't be here with us today in Switzerland. We wrote about how infectious agents can actually exploit the sensory system of the head and neck. So your ears, nose, eyes, and mouth, and then actually invade the brain through those places, causing damage to those places and to the brain, resulting in loss of sense of smell, which is seen in a number of different psychiatric and neurodegenerative conditions. And what we do here is, this is research basically that we're aggregating and harnessing that was already in the literature, and we're bringing it into a high impact infectious disease journal so that other people can begin thinking about it. Also, I wanted to highlight an article led by my friend Momo Vucic at Viome that does microbiome testing. We were not writing about developing any weapons, but this Manhattan Project concept came about because, to quote our team that wrote this, we believe we have knowledge and technologies already available that can diagnose and treat diseases in a very precise way. We need to identify the root causes. We need to describe these strategies. We need to work across the different silos. And then we have a unique capability today to disseminate knowledge due to the technology we possess. And we can also support translation. And one of the things that's really important to me as a patient, because that's how I approach all of this first, is from the patient side of things, is actually getting information, not just in medical journals, but in the hands of people in a credible way, in a broader sense. And we were very fortunate to have very long feature articles in New Scientist, The Guardian, The Economist, and also with my friend and colleague Brian Balin to write an op-ed piece featured in the Philadelphia Inquirer. And, sometimes people will say to me, Nikki, aren't you afraid you're going to scare people if you tell them that infections could cause these diseases? And then I say, I've already been scared when I was sick. And what was scarier to me were 12 doctors saying, Nikki, your syndrome is idiopathic. I detest that word, after being a patient, because giving someone that has neurologic Lyme disease benzodiazepines, does that help them necessarily? I don't think it does. It would have maybe calmed the fear and anxiety that I was feeling, but it wasn't getting to the root of my problem. So we're very passionate about this. And with that, I will say there's amazing momentum and we have reason for great hope. So on June 2nd, which is also my birthday, thank you very much, NINDS, the National Institutes of Neurologic Diseases and Stroke hosted a summit, and they made recommendations for how to invest funds and how to focus research for the coming years in Alzheimer's disease and related dementias. And the number one recommendation coming out of the gates was to investigate how the exposome, things that we're exposed to, influence Alzheimer's and related dementias with focus on microbes and infectious disease. This is groundbreaking and important, and it's indicative of a sea change, in my opinion. But here's what my dream would be. If I were to wake up tomorrow and anything, could happen, this is pretty high on my list. This isn't real. I made it with AI. But I know many of us, many patients, many researchers, many clinicians in the space, advocacy groups have championed for an entire branch of the National Institutes of Health dedicated to infection-associated chronic illness. Because what I want to say is this goes well beyond the brain. Other parts of the body can be impacted by these chronic infectious processes and the host response. And if there were adequate federal funding from the largest funder of medical research in the world, the NIH, we could have some of these solutions in development right now that overcome issues with using long-term antibiotics like I've had to take now for 9 years in order to stay well. So with that, I want to say how grateful I am for everyone here today, for all of our patients, advocates, and everyone that's also on our virtual platform. There are so many people to thank. I'm sure that I will miss something, but know that you are all loved and everyone that made this possible and enabled this. But I want to call out my family, my friends, Dr. Brian Balin, my colleague and friend for 8 years, Dr. Graciela Muniz-Herrera, who we only have the pleasure of knowing because of the linkage with the Osteopathic Heritage Foundation, Jay Feldstein of PCOM for allowing the Pathobiome Research Center to become a reality and for me to have a collaboration hub at PCOM. And I want to also call out our AlzPI teammates, all of our PCOM teammates, the people that help support our research in this event, Steve Sim and Marilyn Einstein. We can't thank you both enough. The Gosnell Foundation, obviously OHF. We have the Tick Boot Camp podcast filming content upstairs and interviewing some of our speakers. So with Ali Moresco and Moresco PR, so we can really increase the scope of how much impact we make today. Thank you to the tech team, Lindsay Kidd, Brian, OU, and always thank you, Charles W. Stratton. You're not here with us today, but I thank you for treating me and making me well so that I could help others. And with that, I'd like to hand it over to Brian. Juicy's presentation. Tom, can I ask you? I don't see the presentation. Where's the presentation of two slides? Ah, thank you.
Thank you all for being here today. And Nikki, thank you for a wonderful introduction. And the thank yous to everybody in this room and to others that are not with us today, that are here virtually or that have supported us over the many years. I'd like to offer more of a historical perspective of what we've been going through for over a century, actually, especially with Alzheimer's disease. But I'm also going to talk about some other diseases that we have to consider as well, as Nikki mentioned, with neurological issues and potentially even whole body issues with chronic infections. So this begins with Alois Alzheimer in the early 1900s, finding a number of cases actually that came to him where he followed them to post-mortem. So he had cases that he had for a number of years, followed them into post-mortem issues, started looking at brain tissues and started finding plaques and tangles and used silver staining to actually identify that. Interestingly, he had contemporaries, just as we have contemporaries now also, that were looking at very similar entities. And it was actually Oscar Fischer in the same year that Alzheimer was accredited with this, what was called morbus Alzheimer by Emil Kreppelin, who was the institute director where Alzheimer worked. Oscar Fischer was actually identifying these plaques, neuritic plaques and tangles as well. And he actually is credited with identifying what these plaques were. Now, at the time, they didn't know that it was amyloid, and they didn't know it was tau. All they knew was that you have these accumulations of material extracellular and also in neurons. What's interesting is all three of these individuals, including Bonfiglio from Italy, started looking at this as an infectious process that, oh, maybe this is syphilis, based on what they saw, especially with these amyloid plaques and the fibrillar nature of that, and thinking that these could be spirochetes, which have a corkscrew structure. And they were looking at it this way. The issue with Bonfiglio was he received tissues from Alzheimer to evaluate in his laboratory what was potentially a syphilis issue, neurosyphilis issue. Well, that's exactly what we're doing today with a consensus approach with all these laboratories of representatives in this room of sharing samples, of looking at different techniques to identify how we can see if we have microbes in brain tissues of individuals dying from Alzheimer's and other dementias and illnesses. What have we looked for and what have we found? Well, here's a very brief list. This is not inclusive. This is just highlighting some of the organisms that are bacteria and viruses, fungi, parasites, complex microbiota from the gut in oral cavities, for instance, that can be involved in this issue. This is why we've now taken more of an agnostic approach of what could be causing the problem, affiliated with the problem, exacerbating the problem, etc. And we need to focus this way because none of us feels that one single thing will actually cause all of these problems. It's more of a multifactorial issue along with a polymicrobial issue. Here is just a brief little summary type of slide suggesting that you have infection and invasion. And I've just highlighted on the left side that you could have infection through the olfactory system, the neuroepithelial cells in our noses, but also through the blood-brain barrier. And we probably have both happening. And this is what we were trying to understand. But in the center of the slide, you see that all these pathways that have been studied and have now identified are involved with the pathogenesis of the disease actually are occurring. And infectious agents can actually impact all of those pathways. And then we have neuroinflammation on the right-hand side. Neuroinflammation is a big key because that is leading then to the generation of damage. And we have chronic damage in this disease and in these other diseases. And that's leading to amyloid and tau and other types of pathology. And by the way, you'll hear from Will Eimer and others that we have those particular ingredients, the amyloids, the tau, alpha-synuclein, even ApoE, the genetic risk factor that is polymorphism, it's not a mutation, can all have anti-infective properties. So there's a rationale of why these things may be accumulating as well in this disease and in other diseases that we're looking at as well. Here we have something I call the new germ theory. And I say it this way because in many ways, chronic diseases and the etiology of chronic diseases actually has already been identified and established. And what we see here are infections that lead to chronic disease. A lot of the viruses leading to a lot of the different types of cancers. Bacteria leading to chronic change. One of them, Helicobacter pylori. Well, that was revolutionary. Why? Because it was found in the guts of individuals, in the stomachs, and thought, oh, that's impossible. You can't have a bacterium growing in the stomach. Well, Warren and Marshall proved differently that you can, and it can actually cause cancer, ulcers, etc., lymphoma, etc. So we know these types of agents do exist in us and can cause major disease. And why aren't we actually accepting that this actually can happen in the nervous system as well? So these chronic neurological conditions caused by all these types of different organisms here, leading to persistency, chronicity, latency. Those are the types of issues going on here. These are not acute infections that are leading to then an acute representation of disease. These are ingredients and organisms that can lead to this type of chronic and persistent problem. These other infectious agents and neurological disease association, I've just mentioned a few here. But for instance, Nocardia asteroides with Parkinson's disease. Well, there are publications that are out there on all of these actually ingredients affiliated with these types of diseases. ALS, for instance. Okay, so there's a recent article, I think today, that came out about ALS might be an autoimmune issue. Well, enteroviruses have been found with ALS. Why aren't we studying that more? Because much of ALS is actually sporadic. It's not necessarily genetic, even though you can have genetic components. That's the other thing that's happened here, is a lot of these diseases do have genetic components where you have mutations of certain genes that are now leading to that disease. But that is typically a very small percentage of the total population that is getting that particular disease. So for instance, with Alzheimer's disease, 2 to 3% of all Alzheimer's disease is due to genetic mutations. 97, 98% is due to something else, not genetic mutations. And this is where our environment is where we are focused on. And that's why these infectious components are so important to understand. The susceptibility of the CNS to infection increases with aging because we say, well, why are a lot of these diseases occurring as we age and in the aged population? Is this something happening early in life? And it could be, but it's manifesting in later age. Well, we have all these different ingredients happening here with aging. The blood-brain barrier, our immune system is changing, blood-brain barrier is changing, cranial nerve insult, for instance, olfaction, the turnover of our olfactory neuroepithelial cells in our noses, basically, will happen at about every 90 days on average. But what hasn't really been studied is, does that actually change? In other words, you get a neurodegeneration and a regeneration process of a cranial nerve or olfactory nerve or cells in our noses. So is that changing as we age? And if it does it make us more susceptible then respiratory wise of getting something into the brain? These are key questions. Neurons changing because of oxidative stress. We know this is happening. Impaired energy production because of mitochondrial damage. And then the neurotrophic factors. Our cells are suffering. They're starting to change. They're starting to be stressed. And we're trying to understand, why are they being stressed? It's not happening by chance. And it's not only happening because we're aging. It's just another thing that is giving us a greater susceptibility. So these driving forces behind the newer discoveries that have taken place and are currently taking place, well, we need to look beyond genetics, the other environmental factors like stress, diet, and smoking. Well, stress, diet, and smoking, that's what used to be attributed to for ulcer generation. And then finding that you have actually an infectious agent here that is actually key. Now, it's not that the other ingredients don't have an effect as well, but you have a key ingredient that actually was shown to be causative. Development of advanced molecular techniques now, all this metagenomic next-gen sequencing, the 16S rRNA gene sequencing, digital PCRing. I'm not even mentioning some of the types of studies being done with expansion microscopy, where you can actually see inside tissues that you now are studying ex vivo and being able to label for organisms. We have a colleague, Or Shemesh, that is an expert in that and has been identifying a lot of different organisms that way. We've used electron microscopy to identify these organisms in brain tissues. Inflammation and its triggers as key components to pathogenesis, a huge key to the whole issue. And the revolutionary findings of Warren and Marshall in 1983. They were vilified when they presented their work in front of the gastroenterologists. They were told they were absolutely crazy. They weren't crazy. We're not crazy either. We believe that what we're finding is key to an understanding of real causation for these diseases. Instead of looking at all these other ingredients independent of infection. Why isn't infection included in the analysis of the exposome? There are key people, epidemiologists, looking at the exposome in this day and age that are not including infection in the equation. It needs to be included. And I'll leave you with this, because I love this quote from Albert Szent-Györgyi, is that discovery consists of seeing what everybody has seen and thinking what nobody has thought. And I think that's exactly where we are and have been. Thank you.
Brian, thank you so much. Absolutely fantastic. And now I have the pleasure to introduce Dr. Mickey Tall from MIT. Sorry, I'm having trouble exiting slideshows today. You can open the PDF instead. For the presenter mode? Yeah. Oh, do you know how to do it? Like, I just. I'm not sure. Maybe I'll zoom in. Ready to do presenter mode with PDF.
I also have the PowerPoint. It just won't, it's too big to load in slides. Okay, perfect.
Thanks, everyone, for your patience. Thank you for your patience. We have some very large files with some very fantastic contents. They are currently loading. Is that Jeopardy that has the song? We need that in the background. Dun, 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 dun. Just kidding. Thank you. All right, so now we can go presenter mode here.
Oh, just enable editing. Okay, perfect.
All right.
Thank you.
All right. Okay. So today I'm going to tell you a little bit about the work that we're doing in my lab at MIT. This is kind of an overview slide of the three main aspects of research that we're really focused on. And so first of all is this concept of predictive diagnostics, which I'm going to tell you a little bit more about. And this is what my lab has an R1 from the NIH to study. And this is a concept that I had started to develop in my postdoc and really kind of built the lab around. In addition to that, there's so much that we're learning from that, that we're investigating in both our mouse model of chronic Lyme, as well as in the clinical study that we run at MIT, which is MIT's largest clinical study and run by our conductor of Maestro, Yuri Kim, who's going to give you a talk in the afternoon and get to tell you more about what we're learning from Maestro about how Lyme and COVID are impacting the brain. Since I feel it important to just kind of give you a tiny bit of background about Lyme, because it's a bacteria that needs to live a dual life between a tick and a mammal. And we are not part of the life cycle of Borrelia, yet we're an accidental infection. And while with diagnosis and prompt treatment, 90% of people can go on to recover. For the last 40 years or so, 10% of people who were diagnosed and treated have not gone on to recover. And that hasn't changed, and it needs to. And the kind of evolution is against us here in that Borrelia burgdorferi is a beneficial infection for the tick. So there's a huge survival advantage for ticks who are infected. And that really drives this ongoing infection in the mammals. And just as a little PSA for you guys, if you think that ticks are easy to spot, some of them are smaller than my freckles. And that classic bullseye rash that you're thinking about, if you're thinking about that target sign, that's often not what a bullseye rash looks like, even if you're lucky enough to have one that's visible. It very well may not have central clearing. So the only thing that they may have in common is that they just continue to expand. I love this slide too, but now I will skip it and I'm going to go to this one. So I also use AI to help me make some illustrations. And I asked Dolly to make me a Rubik's Cube that said infection-associated chronic illnesses. And this was at the very beginning, and it made me this mess. But I loved it because I thought it was perfect. I thought it exactly captured the state of research on IACI right now. So now you know that term. And I think that we're just at this moment where we can start to see how these pieces might fit together. But the problem is that some of the pieces that we think we know are wrong, and some of the pieces that we need to know are missing. And as AI advances, we can start to fill in some of those pieces that, okay, you know what, maybe, yeah, that might be mitochondrial dysfunction. That might be a barrier issue. That might be allergic responses, connective tissue. Oops, sorry. Okay, so this is my immunologist's view of the world in terms of host defense and collateral damage. You have your immune system, and your immune system is tasked with protecting you from invaders by killing pathogens that have invaded and trying to prevent them from replicating. But in doing so, your immune system directly causes disease. The pathogen also directly causes disease. And so I feel like the primary task of an infectious disease immunologist is to figure out, in this context, who is causing the disease. Is it the pathogen or is it the host? Because this is going to be a completely different treatment approach modality. So on the one hand, you may really benefit from antibiotics or antivirals, and on the other hand, you may really benefit from immunomodulators. So at least in the context of Lyme disease and in many other pathogens that we're looking for, which aren't found in the blood and nestle into your body's tissues, we're not going to take you apart to look for them. We're going to do something called diagnostic serology. So you're going to get a lab test, and it's going to look for antibodies against a particular infection. And essentially what we're doing with this technology is we're asking the immune system a yes/no question. Have you ever seen this pathogen before? Yes or no? And I kid you not that this is as simple as counting bulk IgGs of, antibodies against a particular either part of a pathogen or a whole pathogen. Today, we have greater capabilities than asking one question. And so my pitch to you, and I hope I can leave you with this concept, that we can ask the immune system a much more open-ended question. If you saw this pathogen, how did you respond? Was it in a way that was protective, or was it in a way that was catastrophic? And in order to do this, we really have to flip around how we think about the antibody. We have made such incredible advances in how we look at antibodies, but if this is the antibody and this is the area that binds to the different pathogens, we've focused all of that innovation here, understanding which antigens are being recognized by these antibodies. But this is the business end of the antibody. That stem of the antibody is what binds to different immune cells. That is what recruits and turns on different immune effector functions. And we know a lot about these immune cells. We know a lot about these immune effector functions, and they're different. So these different antibody stems, they turn on different things. And so this whole concept of counting IgGs, we're missing out on some really important resolution about the immune response. And I have one cartoon that Nikki didn't have, so I love this one too. And I think that there's this incredible potential here of asking the immune system, because the immune system already saw these microbes and it already labeled them and tagged them with how it sees them. So that information is there, and we just need to be asking the right questions. So of course, in this context, I wanted to share this slide of strep, because as I moved from, focusing my PhD on viral immunity and in my postdoc started looking more into these bacterial infections, I really looked to strep as, something we had gotten right originally. So I thought, wow, rheumatic fever used to be really common. And in the United States, it's become rare because we're so aggressively diagnosing and treating strep. But I now think about this very, very differently. And I think, 20% of people are strep carriers. And yet the way we're doing the decision tree of who we're testing and who we're not, we're intentionally trying to not test the strep carriers so that they're not constantly coming up positive. So if you have strep but no symptoms, we're trying to not test you and not treat you, because you treat this like a commensal and it's just going to keep coming back. But what if you live with somebody who, if they get strep, they're going to get PANS, or they're going to get rheumatic fever, they're going to get these serious, serious outcomes of an untreated strep infection? Shouldn't they know that they're strep carriers and that they have it all the time? And maybe that could change their preventative and protective behaviors. And then, of course, we have the 70% of people who are just going to get a sore throat for a week, and by the time you give them antibiotics, they're already recovering. So I have really changed my thinking about who we need to be watching out for, who we need to be testing, and what question we need to be asking, because is strep there or not, in my mind, is not the most important question. And the most important question in my mind is asking the immune system, how are you responding? And are you responding in a bring the house down kind of manner? And so, this really came to a head for us during COVID. And from the early COVID responses, this is from a review we wrote called Guardians of the Oral and Nasopharyngeal Galaxy, IgA and protection against SARS-CoV-2 infection. But this is around this concept, that we were talking about during the early vaccination strategies, using these intramuscular vaccines to protect your vital organs and this internal systemic response. This is to keep you from going to the hospital. This is to keep you from dying, but this is not to prevent infection, and it never was. If you want to prevent infection, you have to have protection at the site where you meet these viruses, and that's at the mucosa. And that's a completely different kind of antibody response than the vaccines that we were, developing and deploying. And so we had put this commentary out there about how we need to think about different types of antibodies and what kind of antibodies we're trying to elicit and what kind of, what different antibodies do. So this is just to put that for you. There's 5 different isotypes of antibodies. Of these different isotypes, there's different subtypes. And then if we want to get more complicated, which I spared you of, then there's all these different sugar modifications that we can use to further distinguish these different types of antibodies. But what I'm showing you here is that if you're going to get a Lyme test, that's an antibody test looking to see this yes or no question, most of the cases it's just going to be IgG. If it's very early in your infection, maybe they'll look at IgM. But there's all these other types of antibodies, and they could give us this really important infection. And one that I'll tease for you is IgE, because it's this allergic type antibody that we think about. It's mostly made for a parasitic type of response, and it will trigger very different kinds of immune effector functions than the IgGs, and we're not even looking for it. So the assay that I had developed during my postdoc was to take the same bacteria that we were using to infect the mice, the same bacteria that's been isolated in the wild, and to use it essentially like a magnet to pull out antibodies that could bind to anything on this bacteria. And then what we could do is, once we had them on there, now the bacteria itself is our platform. So now that's what we're using to essentially complex everything on that, and the whole assay is running on this bacteria. This makes it extremely malleable for other infectious diseases that we're interested in. And we've shown this across other, fungi and parasites, that this is a way that you can just look at all of the antibody responses against a particular microorganism. And I wanted to give you a sneak peek of what that looks like under the microscope, because this is just so beautiful, essentially, when these bacteria come under attack. So on the left, I think that looks like the kiss, because it looks like they have heads. So these bacteria dedicate a quarter of their genes to defending themselves from one of our immune processes called complement. And so what you're seeing, all those different colors in that head, that's all these different types of antibodies that are binding to that wound that has been created. And on the right, that cloud of red, those are actually bacteria that have been killed by IgM and got zipped up in something we call an agglutination. Oh, and you also saw this like round body of Borrelia in there that were Borrelia really trying to survive even very harsh conditions. But so this is that concept of, what does it look like if we build these illness trajectory maps, and can we use them to distinguish protective from catastrophic responses? And then can we get really, really, innovative and think about, within the catastrophic immune responses, could we now even start to subset different types of catastrophic responses and really further hone in on these maps? So I wanted to show you an example of a map, because you just heard about IVIG, and that's definitely one of the tools in our toolbox. And so I want to show you somebody here who is over a year past their tick bite, got antibiotics, never recovered. And what I want to show you is this person is making a ton of anti-Borrelia IgG1. They probably don't need more IgG1. And I want to show you what it looks like when you're looking at somebody who's on track to recover. They're actually making a ton of IgG2 and 4. And so you see these maps look very different. And I think it begs the question if we could rethink what we're giving in the IVIG, because IVIG is predominantly IgG1, then some IgG2, then a little bit of IgG3 and 4. And I think that looking at these kinds of maps could be very informative for us of could we make just some of the components of IVIG and make it even better? Because what if we're looking at a person who really needs much more boosting of their IgG2 and 4 specifically, but they probably don't need any more IgG1 than what they're already making. And what I was very excited to see is these exact same ratios that we're looking at Lyme, they're starting to pop up in COVID as well. And so this is a preprint that came out very recently, and I'm very excited about it. And they looked at convalescent controls versus PASC, which is long COVID essentially. And you see that they're seeing the exact same thing that we're seeing. They're seeing a lot of IgG1, a lot of IgG3 compared to how much IgG2 and IgG4 they're seeing in people who recovered. And I just want to make one little comment that there was this scandal of sorts around how much IgG4 was popping up with repeat vaccination and boosting in COVID. And based on everything I've seen in Lyme and in COVID, I cannot for the life of me understand why that's a scandal, because from everything I've seen, you want more IgG4. That's a great kind of protective response, actually, to help you not develop these IACs. So just one more look at some of these maps, and the only thing I'm trying to have you see here is that a third of this chronic Lyme cohort from Lyme Biobank has very high levels of IgE against Borrelia burgdorferi, which raises a question I never thought I would be asking of, can you become allergic to certain bacteria? And do we need to rethink everything that we've just bucketed into autoimmunity? I think in the last talk, you definitely walk away from that saying yes. And I think there's another question within that of, if you develop a response that's pathological to a type of bacteria, that to a feature of those bacteria that is shared across many other bacteria. And now this costs you your ability to tolerate those bacteria that lived in your mouth and your gut. And now you're attacking them and you're breaking apart your barriers. And now you have this battlefield happening where you used to just tolerate these bacteria that lived with you. So what do we call that? Because it's not autoimmune. It's not a break in tolerance. You're not attacking yourself. You are attacking something foreign that lives with you. And so I don't have time to talk about our mouse model, but we do have a mouse model of chronic Lyme, and it is teaching us a tremendous amount about these different responses, and particularly this allergic type we find driving mast cells that are driving an infection-induced hypermobility that's also breaking apart the collagen that's holding together the vertebra of the spine. And so you can just see these stretchy spines and a major risk for spinal cord injury. And I was just going to show you one picture from that to say that beyond expansion microscopy at MIT, we're doing label-free microscopy where we can distinguish the bacteria from the host cells. We can see them. You're seeing a cluster of Borrelia up in the left corner, and we call this Borrelia touched a nerve because you're seeing in that purple, you're seeing Borrelia all the way up to that nerve. That pink river running through is a nerve. This is a peripheral nervous system, and I know here we're talking more about the central nervous system response, but there's some really important interplay happening here between the bacteria, the nervous system, and the host response. So briefly to tell you about the Maestro study, this is a clinical study that I'm very proud of, and Yuri's going to get to tell you all of the neurocognitive aspects that we're finding from this, but I just wanted to show you that we changed the name, what the acronym stands for, and this is what Maestro really is. Measure absolutely everything. We have not done enough. And what Nikki was talking about with the systems approach, that's exactly how we're thinking about Maestro. We think that this is a model for systems medicine and that this is what we really need. And we compare people with acute Lyme to endemic controls, as well as people who've had chronic symptoms for over a year compared to long COVID for over a year. And this is a total measure everything approach of, looking at these different neurological, neurocognitive, of deep medical history, physical assessments, hypermobility testing, spine assessments, skin barrier testing, and just essentially every biological sample that you can possibly think of, we get it from them. And I wanted to just present you with this concept. So this woman is in a boat. The boat has structural problems, and it's taking on water, and it's also raining, and her boat is full of water, and we've given her a bucket. So she gets to look on the internet, and she gets to find something that maybe she can pay for out of pocket and try to bucket, one bucket of water out of her boat at a time, but it's just going to keep taking on water. And if we ask her these patient-reported outcomes, are you feeling better as you try this thing? She can probably give us a good idea if it stopped raining, if she managed to get some of the water out of her boat, but her boat's going to keep taking water. How do you ask somebody if their connective tissue is healing? How do you ask somebody if they've stopped these mast cells that were breaking apart the collagen fibers? You can't ask somebody something like that. We need to tell them. And so what I'm urging you is that we really have to measure everything, because the woman on the left may not feel better. She's still sitting in a boat full of water, but her boat is healing. And so, I think there's something really important there. And I just want to mention that, when we first designed the study, we were excluding people with concussions because we were really trying to understand all these different brain features, and we didn't want to be measuring TBI. But what we started to realize is a whole subset of people with Lyme were then having proprioception issues, and then they were falling, and then they were getting concussions, and that was part of it. And so, yeah, part of what we're looking at is a TBI from the concussions, but those concussions were from Lyme because they were falling, because of their proprioception. And it's really made us think about, who are your exclusion criteria excluding? And so I'm just going to get to tell you, just thank you to, you're going to hear about Maestro from Yuri, but I just want to thank everybody in my lab, and just what a special place at MIT. In that upper corner, you're seeing Linda Griffith and Doug Laufenberger's lab, where, we are the Center for Gynepathology Research, and we work very closely together, as well as Sashen Yu's lab, who does all of our label-free microscopy, and in the middle, the Center for Clinical and Translational Research, where we run the study. And I can't wait for you to hear about it more in the afternoon. Thank you.
Mickey, thank you so much. That was incredible. And we're going to have to record a whole nother session so we can catch the back end of that slide deck and get all of that tremendous information. It's my absolute pleasure now. First of all, slight change. Due to a bad cold, we are not going to have Dr. Nagpal here today, unfortunately, but Ravi, we will record something with you, and we will have your talk available at some point. With that, we're going to move to Yves Monet for our next presentation from Drexel. Yves, thank you. Great.
Thank you, Nikki.
Sure, Mike.
Thank you, Nikki, for your introduction. So I will present you today part of our work at Drexel University in the Ehrlich's lab. So I will talk about the profiling of microbial communities in Alzheimer's disease brain. So as Brian told us earlier, for several years now, accumulating evidence suggests that microbial infection could be involved in the etiology of Alzheimer's disease. So as you can see here, there is a lot of work about different microbes, ranging from bacteria like Cutibacterium acnes, chlamydia, spirochetes, or fungi like Candida or Malassezia, and well-known viruses like herpes viride. So this evidence come from large cohort epidemiological studies, from studies of brain, and also this potential involvement of microbes in AD is strengthened by the fact that amyloid beta, which is a hallmark of Alzheimer's disease, is a natural antimicrobial peptide which act as extracellular trap and trapping pathogen and preventing their spread and inactivating the pathogen. So the innate immunity component is important in the Alzheimer's, could be important in Alzheimer's by triggering neuroinflammation. And also there is a link between neuroinflammation and tau tangles, another hallmark of Alzheimer's disease that could be involved microglia cells. So to investigate the link between many bacteria and Alzheimer's disease, we have developed a new bacterial pan-domain microbiome analysis. So first, microbiome DNA is extracted and sequenced. So we use the full-length 16S ribosomal RNA gene amplification, and we take advantage of the long-read sequencing technique from PacBio, which rely on single molecule real-time sequencing. Then we use the circular consensus sequencing or read analysis, and we demultiplex barcoded samples. Finally, we cluster in operational taxonomic units, OTU, and get the taxonomic classification. So finally, we run a statistical analysis based on a differential relative abundance test, which is based on a hierarchical Bayesian model with two Dirichlet, which is adapted from the Dirichlet multinomial model from Harrison, which has been published by Harrison. So our bacterial pan-domain molecular diagnostic assay, first we get a frozen brain from Alzheimer's disease cases and age match control. So the tissue are dissected, DNA extracted, and we run the 16S rRNA amplification. So 16S ribosomal RNA gene contains a variable region interspaced with a conserved region. So the variable region are used to distinguish the different taxon, and the conserved region is used to design quite universal primers to amplify 16S from every bacteria, or more or less every bacteria, because some of them present sequence variation in this conserved region. For example, the chlamydial or the bifidobacterial, the primers are a bit different. So the single molecule real-time sequencing, the double-strand DNA fragment is first ligated with a proprietary adapter called SmartBell to circularize the DNA fragment. Then the DNA polymerase bind to the template, and then synthesize the new strand. So because the circular structure, the polymerase can have multiple passes, so we obtain subreads. And then the consensus sequencing the consensus sequence, circular consensus sequence algorithm allow us to combine these different subread into one read with high accuracy. We are at 99% accuracy and canceling the random sequencing error. Yeah, it's okay. Sorry for the picture. So here is a PyBio SQL system. So in the lab, Josh Earl, our collaborator, have developed a microbiome classification pipeline. So it is MC Smart. So briefly, the sequencing reads are clustered into OTU, and then there is a taxonomic classification of these OTUs, and we get also the read count associated to each OTU. So to test the microbiome differential abundance, we used the hierarchical Bayesian model approach. We use this approach because microbiome data are count data but are compositional. So for each sample, we obtain a proportion of various OTU. And the sum of this OTU rate count, it's constrained by the sequencer. So the read count provide only relative abundance, relative information. So we need specific statistical framework to analyze this data. So we use a Bayesian analysis where the relative abundance are estimated among the samples and the sampling groups. For example, the group Alzheimer AD and the group control. And sorry, the DML enables the detection of difference in relative abundance between AD and control. So first, we want to address a problem which is often evoked in this kind of experiment, is about the potential contamination. If the bacteria are really in the samples or come from environmental contamination. So to do that, for each brain tissue sample, we trimmed the surface of the brain tissue. We call that the edge, and we have done biopsy punches inside the tissue. We call that the core. So the core has never been in contact with external environment, contrary to the edge. So we run the Pendomain analysis on edge and core. We also address the problem of the reagent-related contamination. We assess this control by doing blank DNA extraction control. So we used post-sequencing statistics to identify and to remove the contamination DNA features. So to do that, I used two different R packages based on different math. So one, the decontam is based on the occurrence of the contaminated OTU in the negative control. And for the second one, it removes the number of contaminant reads based on the negative, on the DNA extraction controls. So here is the output from our differential abundance analysis. So because it is based on a Bayesian approach, the result is a distribution of probability. So we subtract to do the difference between the AD and the control, we subtract the distribution probabilities in AD and versus the distribution probability in control. So then if 95% of the difference is close to zero, we can consider there is no difference between AD and control. So we have done this work on two data sets. One data set comes from the University of Arkansas Neurobiobank, and the other data set comes from the Wisconsin Neurobiobank. We do that for the AD group and for the AMC group. AMC stands for age match control, so control. And as you can see there, between edge and core, we can find no significant difference for our samples. So it means that the bacteria we can detect in these samples are not due to environmental contamination, because it is expected in case of environmental contamination, it is expected we get a difference between the surface and the core of the sample. So we obtain the same result for another data set. So it is not related to the data set. We can reproduce this result for different samples. And then we are interested in the differences in relative abundance of the OTU between AD and AMC. So here it is the same kind of output from the Bayesian analysis. For the most of the bacteria, we cannot detect differences between AD and AMC, but for some of them, we can see they are mostly associated with AD samples, and others are mostly associated with control. And in the UMS dataset, we get a high signal for the Cutibacterium acnes. So Cutibacterium acnes is known to be able to trigger innate immunity response and inflammation. Here is the result for the Wisconsin dataset, and as you can see, we have detected also Propionibacterium acnes as mostly associated with AD samples, and other bacteria are mostly associated with control. So we can conclude first, we have no evidence of contamination of the postmortem brain specimens. The Cutibacterium acnes is mostly associated with AD, while other bacteria like Acinetobacter species are mostly associated with the control. The experiments have been done on different locations of the brain, so the frontal, temporal lobes and entorhinal cortex. And we can see substantial variances in the species level bacteria abundance values between these locations. So we will investigate the microbial interaction and their pathogenic impact on the AD brain. We also want to run a high-resolution microscopic technique to spatially characterize this multi-species aggregate. And finally, we are optimizing the species-specific fungal and eukaryote microparasite transplant domain molecular diagnostic based on 18S, ribosomal RNA gene. So I want to thank you all the members of the Center for Advanced Microbial Processing, particularly the head of the lab, Lars Ehrlich, and Michiko, who is a PhD candidate and have done most of the experimental work. Also, our other collaborators in the camp and the neurobiobanks, which provide the samples. So thank you. And just if I can remind, we are collaborating with the Frontiers in Cellular and Infection Microbiology, and research topic for a collection of articles is now open. So if you have some manuscripts that made the scope of the brain pathobiome in Alzheimer's disease and other neurodegenerative disease, so you can submit in this research topic. And yeah, I am delighted to co-edit this research topic with Garth Ehrlich and Lavinia Alberry. Thank you.
And thank you to Frontiers for also sponsoring this event. Now, it is my absolute pleasure to introduce Dr. Madeline Cunningham. And as I mentioned previously, she and her team are the reason that we have a set of biomarkers to market that clinicians can order, called the Cunningham panel, but also now the Brain Autoimmune panel. Moleculara Biosciences offers this. Madeline, thank you so much.
Well, thank you so much for inviting me. And of course, I've seen many of you that I've known from before in the past, and met those of you that I haven't known before. And even you've heard of strep and rheumatic fever already before I even start to talk. So it's wonderful to be here to see all of you and meet you and be able to tell you some more about our work and advancements that we think are important in understanding the streptococcal sequelae, because I'm primarily an immunologist, even though I've always been a microbiologist at heart. So let's see, I need to figure out, I guess, is this?
You can just hit an arrow, I think.
Oh, just on, okay. Oh, good. All right. Just to go over the funding that we've had over the years and my affiliation with Moleculara for my disclosures. We've had funding for years. We had funding for about 35 years from the National Heart, Lung, and Blood Institute because a lot of our focus was on the heart and have been studied not only rheumatic fever, but autoimmune myocarditis in humans. And I have a panel of antibodies that actually helps predict whether a person's going to get better, their ejection fraction will get better in their heart. But it's taken so many years to figure that out. And fortunately, there's a group at Harvard that has similar results, which is really good. It's good for all the humans that are heading toward heart failure, because maybe we can make a difference before they get there and prevent that. So I will also point out we've studied Lyme disease. We've studied many of the diseases that seem to go along with PANS symptoms. But today I'll talk primarily about strep and autoimmunity and infection and group A streptococcal sequelae, and added a little caveat, it's about time. So we'll talk about that toward the end of the talk. But you've heard a bit about the diseases strep causes. And of course, the more invasive disease, I'll point out that right now we have an invasive outbreak in all the countries in Europe. If you've been reading JAMA and some of the British journals, you'll hear, I just came from this spring where I was in Sweden and they had an outbreak of Sydenham chorea. and children were not being treated. So I think people just don't recognize it. In the developed countries, people don't see Sydenham chorea very often. So they're not certain what it is. And also a lot of the children, they didn't even know they had strep. So it's something to think about when you're seeing a child to treat them. So the guidelines for diagnosis of initial attack of rheumatic fever, these are just the Jones criteria that lists the major manifestations. This is through the American Heart Association, and Andrea Beaton is very active now. And she's a cardiologist at the University of Cincinnati in pediatric cardiology. But they keep modifying it. Now they use echo to try to identify rheumatic heart disease. That was a huge argument for years about whether they should do that. But carditis is the most serious manifestation, arthritis the most common, chorea in the neuropsychiatric, and pandas. Maybe should pandas be part of rheumatic fever? We maybe wouldn't have had so much arguing because all the neurologists really believed it was a piece of chorea. And that really is probably true because the antibodies are very similar. And until we could separate them, which is what you'll hear about today, where we could separate pandas and chorea, we believed that the antibodies are the same, but they're actually, in the end, they're not exactly the same. The minor manifestations are in general just for infections. You can see elevated CRP and so on. A positive throat culture, and I think they're getting more rapid tests because someone asked me recently about a new Abbott test that's out. I can't recommend it or not recommend it, but we'll be trying to read about this new test that they think can replace the throat culture, which of course would be a good thing. But until then, I still swab my own throat when everyone in the lab has strep to try to be sure I don't have it. And then elevated streptococcal antibody titers. This is your ASO and anti-DNase B that are important in using to detect if a child has seen strep. It will not tell you if they have strep at the time, but it will tell you if they've been exposed within the last few months. And children over the ages of 5 to 15 build their immune response against strep. And that's why during those ages is the peak of rheumatic fever, the peak of pandas, and the peak of the sequelae is because the antibodies actually occur over time. It's about time, is that when you begin to have strep infections, you don't immediately get rheumatic fever or immediately get PANS and PANDAS. You have them over your childhood lifetime. And if you're one of those people that's susceptible either through HLA or we have certain biomarkers that are, this biomarker is an IgG2, that when we look at all rheumatic hearts, they are covered with IgG2. But the specificity is for the GLUTNAC epitope. And this is just a piece of the group A carbohydrate. And our vaccine studies in the lab of the whole group A carbohydrate doesn't really show cross-reactivity. But the problem is that using that epitope as a haptene, we can pick up the cross-reactivity. And this is with the sequences of the M protein and other proteins that may have alpha helical structure. And the reason is because that alpha helical structure cross-reacts with all those GPCR autoantibodies that cause all of these types of infections, some of which you study. So we'll move on. Just again to reiterate, we'll be talking about the brain sequelae. Oh, I don't know if my video is going to work or not. I think I can't do it. I'm sorry. I was going to show you a video. I can show it at the break. I can get, except I didn't bring my computer. I usually do, but it was just heavy and I didn't bring it. Anyway, this is a diagram in one of our reviews showing how strep affects you at the pharyngeal epithelium. And as that happens, it's getting up in your sinuses because it's just like staph. If you have it on your skin, you have it in your nose and you have it in your throat. And so it's going up to affect the Th17 cells in your in your sinuses. And this is where the rubber meets the road, because it's going to affect the cytokines. You're going to have IL-17A, and this is going to break your blood-brain barrier and allow antibodies or anything else and itself to get into the brain and affect your microglia, affect your basal ganglia. And that's, of course, what happens in not rheumatic fever, in pandas, which I wish rheumatic fever, and maybe someday they'll change and accept it. And if that's the case, then we won't have to worry. Children with PANS and PANDAS will be treated, or maybe not PANS, but will be treated with the same type of treatment that you get for rheumatic fever, which is until you're 21 or so, you're treated with antibiotics. So B and T cells are generated that produce cross-reactive antibody, leading to this Th1, Th17 response. It is really destructive in the case of rheumatic fever, affecting the heart, brain, joints, and skin. And so streptococcal-associated behavior and movement disorders, we won't focus on the heart or any of those today, but in Sydenham, Korea, early neuropsychiatric symptoms, just like pandas, happen. And you have mild coordination or severe disruption of movements, and that's the frank Sydenham chorea itself, because the chorea is the involuntary movements. The neuropsychiatric symptoms are the tics and OCD, but you can see in Sydenham chorea, but these children actually are so affected that the parents are more upset because they can't write and they can't walk. So they don't even think about them being neuropsychiatrically affected. OCD, hyperactivity, tic-like symptoms, and then in the case of these diseases, fine piano playing movements of their fingers and toes that actually can help you with the diagnosis. So the antibody cross-reactivity between group A strep and brain antigens, the M protein and group A carbohydrate are such well-known antigens on the surface of the strep. The M protein, this alpha helical amphipathic type molecule that's able to bind these GPCR autoantibodies because of its sequences that are hydrophobic. And then the group A carbohydrate shares some of this in its glucanat molecule, the N-acetyl beta-D-glucosamine. It's its main epitope that's repeating. But the whole group A carbohydrate by itself, we have not studied that because it's too hard to get it purified. And the antibodies actually bind there on the M protein. And you can see this was a figure that I can't take credit for, that was in the Nature review on group A strep and the sequelae. And so this is the diagram showing the different antibodies, lysoganglioside, which actually is more prevalent seen in Lyme disease situations. And Lyme and strep are very, very dangerous. They cause a much more serious disease when you have the two of them together, because they're both such activators of Th17. And then the abnormal movements are seen, and this is because of a dopamine receptor encephalitis caused by the autoantibodies to D1 and D2. And what I'm going to try to explain to you is that D2 will go with chorea and D1 will go with pandas. And so they're completely different diseases, but they still, I think, belong in this category. And you can see the basal ganglia, and that it's important that children are treated, and that they are identified. I wish we could identify them. If we could ever get our biomarker together, we could identify those people that should be watched more carefully and treated more often for strep. Of course, I would never not treat myself for strep. I always get a panic because I have valvular heart disease, and I don't want it to be any worse. And I think it maybe comes from a lot of even the valvular heart disease that they don't classify as rheumatic, that you probably got that from infections. Abnormal immune responses, that if we can figure out all the immunomodulatory therapies, IVIG, rituximab, in very severe cases, antibiotics and steroids and less, so that we'll have no pandas and no tics and OCD. The criteria for pandas: presence of OCD tics, prepubertal onset, episodic course, association with neurologic abnormalities. There may be some choreiform movements, but those are not present in all pandas children, only the ones that have the small piece of Sydenham chorea with it. And those have some mixed antibodies of D1 and D2. A temporal relationship between symptom exacerbations and strep infections. And here's a big list of the different diagnoses. Because of time, I'm just going to move on. You can have your chance to see them for a moment. A normal child would be able to label and draw a microscope, but a child that has pandas or the problems with the involuntary movements, which is the piece of Sydenham chorea, is going to not be able to write. So MRI evidence is positive with the caudate and globus pallidus, and the caudate and putamen compared to the globus pallidus and the thalamus. And you've seen some of these. These are old, but just for those that may have never seen them, the good effects of IVIG, and this was the Perlmutter study. And then in the plasma exchange, it was also very effective. And you can see here the staining on the left of chorea and pandas, how they look similar. This is probably staining perhaps of the tubulin, because that's the other antigen, tubulin lysoganglioside D1 and D2 receptors, and then a CAM kinase signaling of a human neuronal cell. And then the controls are negative. And then this is the example we used in our article when we first came out, was the Sydenham chorea human monoclonal antibodies as they bind to the surface of the human neuronal cell line SK and H, and they signal CAM kinase, which all of you have seen these, the CAM kinase being highly elevated in Sydenham chorea and moderately in pandas and not in non-pandas. And that the antibody, this binding to D2 drives the dopamine production, so you make too much dopamine. The D1 actually enhances the receptor and makes all that dopamine worse. So this will show one of the most important experiments I think we ever did that took about 10 years before we published this paper, the transgenic expression of a chorea antibody from humans in mice. And so what we found is that I don't show all the tissues, but the hippocampus wasn't affected. And there were a few of these in the cortex. Maybe they were pyramidal neurons. I don't know. I'm not a neurologist or even really much of a neuroscientist. I'm more infectious disease and immunology. But you can see the co-localization in the mouse brain from the B cells were producing this antibody, and it actually deposited in the basal ganglia, we think in the substantia nigra or the ventral tegmental area. And they were stained with tyrosine hydroxylase antibody to show that they were dopaminergic neurons. And then the, I know there must be a pointer, let's see. I don't know, maybe not. But you can see the TH antibody that stained the dopaminergic neurons and merged with the fluorescent, because we put a fluorescent tag on the antibody we put into the mouse. It was on the gene so that it came out and expressed a marker that we could identify only that antibody. So that just shows that the antibodies do get into the basal ganglia. And so is there evidence for a role of the dopamine receptors in Sydenham chorea pandas, and that dopamine receptors present on the surface of neuronal cells react and respond? And actually, you could see they get into the cells and spare the nucleus. So they're gumming up the network inside the cells. And so it takes time for children to get better because these are inside the cells and have to, because chorea has never killed anyone. And I get it, you know, people don't want to overtreat either for strep, but you want to be sure that you're not neglecting people that might be getting these diseases. So in studying the signaling, you can see what we've shown before, that This particular monoclonal that deposited in the brain signals, it's a D2, so it's inhibitory, and so that's why it goes down, and you see the dopamine go down. The response is I can't think of the word. The response changes as the concentration elevates. You can see that you get more and more response, and so it's good. The Sydenham Chorea sera then actually behave just like the monoclonal antibody from the patients that we made the monoclonal antibodies from, and they get better. The antibodies go away during convalescence. And then for D1, this is our latest paper where D1 was found to be the antibody to target the D1 receptor in pandas, published recently in the Journal of Clinical Investigation Insight, where the dopamine receptor autoantibody signaling in infectious sequelae differentiates movement versus neuropsychiatric disorders. And Dr. Chandra Menendez was the senior postdoc, and she's out looking for a assistant professorship now. And then this is just old, but I wanted to show it before I start showing her data, is that we always believed this was what had happened, but we couldn't prove it. And so that's what this study did, was it proved it, where pandas were always highly significant for D1, even though there was some in Sydenham Chorea sometimes. Sometimes there's not. And that it also was present in Tourette's. With OCD, present in Tourette's with OCD. So the pandas D1R monoclonal antibody, instead of making too much, causing more dopamine to be produced, because I didn't show you all of that past data, you can see that it enhances the dopamine signaling in the blue. When dopamine's added, the signal is enhanced in the presence of the antibody. This is just a monoclonal antibody from pandas for a patient. And then the same here where there's reactivity, and it just simply shows you the GPCR signaling curves where we have the gene blazer transfectant that's expressing D1 only. And so it shows that D1 is signaled by these autoantibodies. And we did this with sera also. I just don't have the kind of time to show you everything, but okay, well, anyway, I still have a few more to go through. But the D1 receptor, we used siRNA to inhibit the signaling, and so this abrogates the signaling. So it was D1 that we were actually looking at. And the distribution, this is in a study, not ours. Well, we published it, but it was a study that came from Israel. Dr. Hilla bin Pazi, a neurologist there, that actually believed in our work. And we're so grateful to her because her best friends were those neurologists here in the States that we always would, I worked with Harvey Singer for years and years and years. And so when our paper was finally published, his wife said, can't you think of anything else to work with him with? I miss him running around the house screaming about you every night. It was pretty funny. Harvey's a good friend, but he's a leading pediatric neurologist from Hopkins, and we worked together and published together. But he didn't always agree with everything I said. So, but the distribution of anti-D1 and anti-D2 in her cohort, because in her cohort it's mixed, but in some cohorts it's not mixed. It can be all D2 and very little D1 in Sydenham Chorea, but it can be, and we showed all 4 of these cases in the paper. I just am not going there here. But in her cohort, there was D1, which is on the left, and D2, which is on the right. They were both significant. And it correlated, if you looked at the ratio of D2 to D1, it correlated with neuropsychiatric symptoms. And so here it is, it's about time. And this is a hard set of graphs. We haven't published it yet. We keep working with this paper to try to make it as good as possible because we want, of course, the AAP, the American Academy of Pediatrics, to like it. And so we're trying to get it ready to submit to the Journal of Infectious Diseases. And I'll ask you to follow those that are the, I don't think I have a pointer, but they're the white ones. The white ones are only in the beginning, except in the ASO, because ASO follows their strep titers. This is a strep titer. But the autoantibodies, you can see that it's only early, and then the others continue to elevate. So what happens is that within the first 2 weeks, you make these antibodies, but in normal people, they go away. And then in people that get chorea, they just continue on up. So that patients, I don't treat patients, but I do consult them and try to help them find care, and also try to help them understand the panel and understand what maybe they can do. And I have cases that I have seen for long years. I have one case that's had Sydenham, had a chorea, that has the D2 antibody. She's 74, is Ashkenazi Jewish population, which is the highest Sydenham chorea. And that's what this study by Dr. Ben Pasi found. They had the D1 and D2, both of them. In some, it's very high D2, as you can see here, that it's the dominant antibody, and that is what is the characteristic of Sydenham chorea. And again, just the same diagram showing that too much dopamine is being produced. And of course, dopamine is the problem in Sydenham chorea. It's a central dopamine issue affecting the basal ganglia. And so then just to close with some of the other, the NIMH clinical trial that was run most recently that Kyle Williams published he was at Yale at the time, and I think he's at Harvard now. So Kyle is a good friend, and Dr. Jim Leckman that he worked with. You all probably know all these people. The autoantibodies are elevated in PANDAS and PANS study subjects participating in that. This is the NIMH study. And you can see those that recovered, the antibodies went down, all of them except tubulin. Tubulin, I think, is more related just to the infection itself. And so the others are more related truly to autoimmunity, where you're developing this and it's getting into the brain. And you can see for all of them, the D1 and D2 and the lyso, and then the CAM kinase, and for all antibody tests combined, they are reduced when you recover. And so what we did to try to better understand whether or not these were biomarkers, what Dr. Menendez did was she decided to use ROC curves, and receiver operating characteristic curves, which are graphs that evaluate the performance of two characteristics or binary classifiers that plot the true positive rate, your actual positivity, against false positive rate, and it visualizes how well a test will distinguish between positive and negative cases. And so you plot the area under the curve. And D1 in Sydenham chorea, even if you have it there, has no predictive value, whereas the D2 has great predictive value. So that's why we were doing this. We did this in pandas, and I didn't have it. I don't have it separated to bring you. They're all four of them are shown in a big figure in this article. So you can see on the right the predictive value of using your test. I mean, if you want to find out, you can always treat children based on what you think it is, but to get a true diagnosis. And also, potentially, we'd like to get the antibodies into the formulary of the IVIG so that insurance companies will pay, and we want to try to work on that. And I know legislatures have recently, there are 10 states that I think have edicts that they have to pay the insurance now for the IVIG. So what we think is there's a spectrum of behavior and movement disorders. And we've also studied this in autism, but I can't really bring you all those results yet. One, I don't have time, and two, we haven't published them, but it's similar when they have pandas. Some autism is not autism, it's pandas. but other times they have it mixed together. We believe that Sydenham chorea is on one end of the spectrum and that OCD is on the other end, and that in between you can have patients that have both of the autoantibodies, but the D1 is causing the neuropsychiatric symptoms and the D2 is causing the movement disorder. And that these are, of course, reacting with these receptors and leading to the basal ganglia encephalitis that is caused in the basal ganglia. And that D1 is now a biomarker for pandas and D2 a biomarker for Sydenham chorea. And the model, of course, is binding of high affinity cross-reactive antineuronal autoantibodies that may cause alterations in dopamine neurotransmission, with the hypothesis that antibody-mediated increases in central dopamine levels and selective activation of the receptors. With D1, now we know that when you put dopamine with the antibody, it's double. You get even more activation. And so in patients, our D2 has finally made the list in Lancet's recent position paper on autoimmune psychoses. D2 made the list. And I think D1 contributes, especially when the two of them are so high. When we see 32,000 or 64,000 for D1 and D2, these children are psychotic. The cases are very difficult. This lady, this 74-year-old lady, she's not psychotic, but she is choreiform all the time. It's really, really difficult for her. And so finally, she's gotten over her paranoia to some degree, taking olanzapine. And so she has finally, after 4 years, going to get her IVIG. I mean, it's just been horrible. If you see these cases and you treat these children, you know how awful it is when a child won't take treatment. I mean, I get these cases where I just visit with them. I was telling the Manfuls, because Dr. Manful is like a therapist. She has her PhD in social work. And so I feel like sometimes I should have done that too. Or maybe that was my calling, to talk to everyone for the rest of my life. But anyway, this combination of antibodies doesn't help you, and it produces altered movement and neuropsychiatric symptoms in Sydenham chorea and pandas. And just finally, I know you went, will I ever shut up? That the disease symptoms are a two-step process, and we have Dr. Agaloo all these years that we've worked, not with him, but we're hoping in this funding crisis we're all in, that maybe we were going to work together to try to improve our funding situation. So we're actually going to look at cells and tonsils with him for the IL-17A and do some single cell sequencing. So immune responses and opening the blood-brain barrier are very important. And he and Dr. Cleary, Pat Cleary was a huge strep person, geneticist in Minnesota, and they worked together to show that strep did this Th17, IL-17 in the sinuses and broke the blood-brain barrier at the olfactory bulb, where then the antibodies would get into the brain. And so I have to thank Dr. Sweeto too, because without all of her samples, we would be nowhere because they were so meticulously collected. And we thank many of our funders and generous donors to our own OU Pandas Research Fund and the David Judah Fund at Harvard that helped pay. This was the late Dr. Michael Janicki, who died recently, and gave us $100,000 to study 1,000 people so that we could have these biomarkers that you are able to use today. And my laboratory with people that work on the heart and brain autoimmunity, including Dr. Menendez. And then Dr. Christine Curvan, who was so instrumental in helping develop the panel, is a full professor at California State University in Sacramento, and has been there for years. And, fortunately, in their 60s, she just lost her husband, so it's just very sad. But wonderful that she still contributes to our program. And then the group of people that have helped for so long. Dr. Cox, who did the transgenic mouse, that was an amazing feat. and Dr. Joel, who did the animal model. I didn't even mention Dr. Remberg, that's with Betty Diamond in New York at the Autoimmune Institute there, and Yale, the group there, and Dr. Ben Pazzi, Dr. Fallon, Harvey, and my statistician, and of course, the late Ed Kaplan that all of us remember, strep expert at the University of Minnesota, and such a great part of the AAP. That's it. So thank you so much. And thank you for the extra time. I probably bored all of you to death.
Yeah. So we're going to do the Q&A now. And for our members that are joining us virtually, what I'll do is utilize the mic. Can we turn this one on, or should we use the mics that are in front of each participant so that when they ask their question, the Zoom members can hear? So what would be great if you turn your little speaker on in front of you, then wonderful.
Distribution studies more prominent video rather than the reason I asked is about 12 years ago, one of our 12 grandchildren developed pandas in the Northeast Ohio area, and sort of, you know, the medical community said, you know, what's this, and his father, who's from Finland, was talking of, I don't know whether that's coming through or not. His father was talking to his father in Finland about this manifestation, and his father in Finland said, well, you know, it's Panda's disease, like it was common knowledge in Scandinavia. And it seemed rather strange that they would be so familiar with this. If there are geographic
Well, I can,
there are geographical distributions that you know of, other than I suspect in Scandinavia, they seem to be very familiar with it.
well, I can tell you first why they're so familiar with it in Scandinavia is because they are meticulous. Their CDCs in Finland and Denmark particularly contacted me. I mean, before I hardly had taken a breath and known what pandas was, and they were on it and collecting samples from all their patients in Denmark, and I think they were doing so in Finland and in Sweden. The Scandinavian countries have this amazing effort that they collect from their populations constantly. And so they were very well aware of all of this. And there was one person that I worked with in Denmark. He's gone now. I think he actually had a brain disorder, maybe not Alzheimer's, but something similar, that he died quite young, and it was very sad. But anyway, they had all of that out there on the internet. I mean, everything I said, it was almost like it was out there, and they would talk about pandas from Dr. Sweeto too. I mean, it would, in the CDC here, even though they were thinking about it, it was not. There's a lot of consternation because of some of the neurologists that felt that it should be Sydenham chorea. Of course, I can't understand all of it, but it seems somewhat better now that we understand more of the science and the mechanisms of the disease. But I can also say that I trying to collaborate with a lady in Mozambique, and she is a cardiologist, but she says she has Sydenham chorea and pandas there in Africa, in Mozambique, just, you know, the same as we do here. But of course, they may see more of it because they don't get their patients treated as often. You know, this is the situation in the African countries. Africa and India and China all still have more rampant sequelae than we do here because our children get treated frequently for strep infections, and so it's less. Every time you have an invasive outbreak, which we're under right now, it's at least double what we've had before. The invasive strains are going around, and this has been found in Sweden. I mean, see, they were on top of it. I was just there in April, and all the epidemiology, the people working on it had everything under control. They were able to show it at the meeting where I attended, and So I think they are first to know. And England had a big outbreak that was written by the people that are in the quote CDC in England. Just all this is published in the 10 states that had strep in the U.S. is published. That was in JAMA this summer. So it's here again. And we had our first case of rheumatic heart disease in Oklahoma City. They called me this summer. So it's out there. I don't think there's one, when you have an outbreak, yes. Utah, remember in 1983, we had a huge rheumatic heart disease outbreak. George Vesey was the cardiologist overseeing all that. We went and actually tried to get money from NHLBI, but we never could get a program project grant for that, even though they sent 35 people up there to review us. Then we never got any money from it. But that was a focus, so yes, it can be geographic if you have a particular outbreak. We never understood why it was just there.
Well, it could be that it shows up more in the Scandinavian countries because they have such a good recording system. I was over at
Exactly.
studying at the Neurohem at Karolinska, and I was amazed at how they track their patients. So I suppose if you did studies of incidents in that area, it would show up more because they keep records of it. So it might not show up in some other place like Afghanistan that they don't.
Right, but I mean, amazingly, in Mozambique, she said, yes, we have those here. I can send you samples from all sequelae. So we're still trying to get the money to get her samples from her residents because we have to pay them.
Thank you.
Well, you're welcome.
Is the mic on? Can you hear me? We will also take a brief break soon as well to give everyone a bio break. But Herb, I see you have a question.
This is more of an answer. In psoriasis, which is related to strep,
You have one paper on psoriasis.
I found biofilms of strep in the tonsils, not in the skin.
Yes, absolutely, because the antibodies are to keratin and they bind in the skin and cause inflammation and psoriasis.
There is no psoriasis in Lapland, the northernmost part of the various continents. And there wasn't any psoriasis in some of the Pacific Islands. The Big Island, Australia, didn't have any psoriasis until the English prisoners were sent there. So yes, there is specific places.
Let's see.
Does anyone have questions for any of the other speakers this morning as well?
I have one thing that I really need to say that I forgot to say when I was talking is that it takes repeated you probably heard, maybe I said it, but it takes repeated streptococcal infections in order to get sequelae. You don't just get them from one infection. So they happen over time. That was why I had It's About Time. I wanted to be sure that was clear because it's something that people don't realize when we use our animal models and we give them injections of strep. It takes 3 or 4 times before you see changes in the heart and you begin to see valvular heart disease, before you see changes in the EKG for the long QT. I mean, we have a model that actually looks like rheumatic fever and looks like Sydenham chorea. They even have the movements, but it takes several immunizations of these materials in order to get it. And we feel that in people, it takes a number of infections before you're going to see disease. So anyway.
Good morning. I'm Mario Grijalva from Ohio University in Athens. I have a question for a previous speaker or for the panel in general. In one of the introductory slides, there was a mention of parasitic infections as they relate to brain health and the subject matter here. And I have worked for many years on Chagas disease.
Oh, yes.
That was not listed. Toxoplasma gondii was listed, and I would like to ask the panel, what are your impressions or regarding Chagas disease and brain health? Thank you.
Well, it's an issue in, like, for example, the working group at NIH on myocarditis includes Chagas disease. I mean, because it's part of the picture, but it's a group of patients that have been infected with the parasite, and it's in the heart. So you've got that, but that is exposing myosin in the heart, and cardiac myosin is the dominant antigen. And when I said we have a biomarker that actually will determine if a person's heart is not going to do well over time. The cardiac myosin's exposed when animals are given cardiac myosin in the right HLA, those animals will develop myocarditis. And so the same thing for Chagas disease. You have the parasites, you expose the myosin, and those people that will have the right HLA, they will get even all the autoimmune myocarditis in that situation. And those are autoantibodies to cardiac myosin. They actually cross-react also with the beta receptor, which we just learned that is how the antibodies trigger fibrosis in the heart, because they deposit all in the heart. Other cells lead to fibrosis in the heart too from all the damage, but the antibodies can initiate it. So does that answer your question? Did
Thank you.
so.
So what we'll do now is any other questions we can reserve for a nice slot that we have at the end of the day for a panel discussion. And we'll take a bio break and reconvene back here a little bit before 10:40 so we can start back up again with more presentations. And then I'm also going to put up on the screen in case anyone that is in the Zoom or here has questions about accessing care for some of these conditions. I'm going to put up some resources that you can access, looking for clinicians specifically. You don't mind helping? Yeah, that's great. I'm just going to actually.
There's small tissue here on my eye, so he's just vector-borne infection. I practice. I have that.
Hi, everyone. We'll get ready to start up again here. If everyone could take their seats, that would be amazing.
Minimize this. Minimize this.
I'm sorry, thank you.
This.
Oh, perfect. Do you mind? Can I ask you a favor? Would it be terribly inconvenient if I had you come sit closer to keep things smoother? Because I feel like I'm not really very good at this. Thank you. So this is, oh no, we have, here, we need this one.
It's the blue one.
Sorry about that. Okay, cool. Sorry. Are we, and our Zoom is still,
Still going.
okay. Thank you, everyone, for grabbing your seats. We're so excited to have our next speaker, Dr. Susan Manful. So if everyone could grab a spot and settle in, we'll get started with the rest of this particular session. This is extra special in light of Dr. Cunningham's presentation, and it's just an absolute pleasure to bring Susan here, who has been an unbelievable advocate, and their foundation, not only, embracing and bringing forth the memory of their daughter, but also helping families all over the place and also helping to spur research. Susan?
Hi, everyone. I feel really fortunate to be here talking to all these smart researchers. And I imagine online there are some practitioners and some friends. And I feel really fortunate to be here. I think that you all are in a place to have a real impact on the trajectory of this disease. PANDAS, which is part of the larger, broader category of PANS. It is an infection-associated chronic illness, which I know is a term that you've already heard used. And in our case, I'll reinforce the immune-mediated part of our disorder. Quickly, I'd like to say a huge thank you to Nikki Schultek and Brian Ballin for organizing this and for inviting me and the Alex Manful Fund. Now, Nikki asked me to share my story, or to share Alex's story rather, and what led us to set up the Alex Manful Fund. What is our mission, and what do we feel are the unmet needs of this field? And that's a lot to cover, so I'm going to dive right in there. I'm going to dive in with some urgency too, because people are dying from this disorder. I know full well, and as does my husband. This is my husband, William Manful. He is the co-founder and the tech man for the Alex Manful Fund. But I do speak with some urgency. We really need to understand this disorder more and understand how to identify it earlier, how to treat it more effectively, and ideally how to prevent it. And I also speak with urgency because every day I get an email from someone, at least one email from someone, usually a parent, sometimes a patient themselves. And today I got one. I wish that I had that person's permission to share it with you, but just underscoring that there are not enough doctors, there are not enough knowledgeable practitioners in general who can recognize this disorder and diagnose it appropriately. So let's get started, and let's begin at the beginning with Alex Manful. Alex was born in September 1991. And at the risk of getting too personal with you, but I feel it's okay because I've told this story so many times to other people, that she arrived 2 weeks early. I was tidying up after a ladies' lunch, so to speak, and my water broke. So I called my husband and I said, yeah, she's coming. Though she didn't emerge for another 18 hours, 12 hours of which was serious pushing on my part. And it turns out that she was in the occiput posterior position, which if you're not a medical person, I'll just refer to it as the sunny side up. So it was a difficult delivery. And I bring this up now because, again, we used to joke about it, but that was the first example of Alex doing things her way. And she would continue to do that for the next 26 years. And I think that that personality characteristic may have contributed to her demise in that she thought she could handle this, she could tackle this. So we loved her immediately, as you can see there, and she changed our lives in every way. We were a very close family. And I was especially close with her because my husband traveled quite a bit, so I had a lot of time to spend with her. I really would like you to know her a little bit. And here she is in her various poses when she's young. She was really funny. She had a very contagious laugh. She was curious. She would, I think, really observe things during the day. And then at night, when I was putting her to bed, she would ask me her questions. Like, why can't God fix an artist friend of ours who had cancer and who was dying? She had great imagination. I would see a box and I would think, okay, maybe I could put a gift in it. And she would say, hey, I could make a computer out of that. And she made a keyboard and a screen and so on and so forth. She was very creative. Of course, she was an only child and she needed to be, but she had villages of, do you remember Beanie Babies? Well, she had every Beanie Baby possible, I'm sure. And she had villages in which the Beanie Babies lived. And her dog, which you'll see at some point there, was also part of that village. She had a lot of charisma, but she was also very quiet and reflective. I think she was kind and she was thoughtful, particularly with her animals. And you can see there in the photograph down on the bottom on the left-hand side that she was drawn to scary things. Whereas most little girls might go to bed with a doll, Alex went to bed with Jack Skellington and two rubber skeletons. She was very different in that respect. In fact, at an American Girl doll tea, she took Jack Skellington. So she was in more detail here. I wanted to tell you about some of the qualities. She was a great writer, and from the very early part of her life, she was writing. She won 4 Reading Rainbow contests, and in fact had one of her articles turned into an animation, which you can still watch on Zoom. She also, after creating many newspapers on her own, created a newspaper that lasted for about 6 years. And it was called the Animal Tribune. And people in Portsmouth still come up to me and talk about the Animal Tribune. So, I mean, in other words, she was very organized. She could really get things done. She and could keep her girlfriends on track. There were a couple of boys every once in a while, but she said boys just couldn't do it very well. I mentioned this egg hunt that they had, and that they had an egg hunt for dogs to raise money for various animal charities. And I sat down with the group and I said, Alex, this is a great idea and you guys have worked really hard. And if only a family comes, that's okay. But it turned out that there were like 60, 70 people who came, and a man brought his goat, and the police came because we should have had a permit. But when she wanted to get something done, she got it done, and she believed in herself. In the process of writing, she also got a press pass from the Portsmouth Herald and went to Westminster. She was an artist. I chose these two photographs, but I could have chosen any number of them. She used pen and ink and watercolors and oils and was a very prolific artist, so much so that we turned some of her photos into note cards, and she had a little business. And she was also asked to create images for various organizations, and here we have one from the Ogunquit Playhouse, with Annie. There's another example of winning a contest, and her drawing was on the back of that trolley. She was an athlete. She was an avid athlete, a tri-varsity athlete in high school. From first grade, she played soccer all the way through the end of college. She was a runner. She was a squash player. I didn't put that picture in there. But she was also on the rowing team beginning in high school. And she was a coxswain. For those of you who don't know what a coxswain is, it's that tiny person who sits in the back of the boat giving orders to, in her case, she worked with men. So it takes a certain personality to be a coxswain, and she had it. She could command authority, but she was also really likable and very knowledgeable about how boats worked. Here again are her various animals. The snake actually wasn't hers. We were visiting a friend who has a zoo of snakes. So I'll leave this one on there because eventually she goes to Princeton and graduates. I can't cover her life in as much detail as I've been doing right here, but I did want to give you the idea of how grounded she was. She had no history of psychiatric disorders, and she had a really unremarkable medical history. No autoimmunity in her life, or well, until we came across pandas, but no other autoimmune disorders, I don't have any autoimmune disorders that I'm aware of. My husband, I think, probably has one of them. But she was really well grounded, organized, et cetera, et cetera. All right, so before she went to Princeton, and she actually was recruited by several different top universities for her coxing and decided to go to Princeton. Just before she went over to Jordan to study Arabic. She already spoke fairly fluent French, and she could write Latin, and she wanted to get Arabic underneath her belt. She had a great time, although I will mention that she was assaulted there, which caused a fair amount of trauma. She was okay. She was okay because she had the wherewithal to say to the man who had chased her through the hallway up to the door that she was trying to get in, that Allah would not like that. And that upset him so much that he stopped trying to put his hands underneath her shirt, and she was able to get away. But she did have to go to court there, or chose to go to court. And again, to her personality, we said, forget it, just come home, which would have been okay. She was there on a State Department scholarship, and they said it's fine, but nope, she wanted to follow through. So she learned a lot, but she also came home with strep. And that was the first time that she had been diagnosed with strep. So, Mickey brought up strep carriers, and I don't know, maybe she was a strep carrier because in spite of all the carpooling that we did and all the people that she knew well who had strep all the time, she never got strep. I have only had strep once, or at least been diagnosed with it once, and on that occasion, the hospital called me and said, no, you don't have it. And then they called shortly back and said, oh, you do have it. So it was kind of unclear to me whether I did or I didn't have it. And my husband also has not had strep, at least according to his parents. So she was given antibiotics and things seemed to be, fine. And she was off to Princeton. Everything started off well, but in her sophomore year, when she was 19 years old, she had a very bad sore throat and wanted to go to the health center, but the coach, her rowing coach, wouldn't let her off the water. And it's hard for people who are in year-long sports like that, in which they are on the water, in the case of rowers and coxswains, for 5 to 6 days a week. But she finally got there. She was very sick. And she also was notified that she was just getting over mono, though perhaps probably EBV. She was given 2 rounds of antibiotics and all seemed fine, but of course it wasn't. And she was still in the health center. I'm going to mention some details here so you really have an idea of this disorder, at least how it affected her. She was allergic to sulfa, quinoline, and penicillin. She also developed psoriasis, and I can address a couple of those questions there. Eventually half her body was covered with psoriasis. There's not a history of psoriasis in our family. And she eventually took a biologic which more or less treated it. That was Humira and then Cosentyx. She had acne, which she had never had acne before. And that's an important point as far as her history goes. So shortly after this bout with strep, and again, I didn't think anything about it because I had just grown up with people and observed other children who would get strep and they'd be perfectly fine afterwards. But she said to me on the phone, Mom, I get really upset when the path from one place to another on campus is blocked. And she said, I have to go out of my way and come back, and it's just really frustrating. And I thought that was a time issue, a stress issue, because all colleges are stressful. Princeton is particularly stressful, and she was a full-time athlete. I mention that here because being under stress would be the most parsimonious explanation for what she told me. Had I had my psychology cap on, I would have asked her, well, tell me a little bit more about that. And maybe it would have come to light then that she was counting steps, because it didn't come to light it didn't come to my attention for about 5, 6 years. And she was surprised she hadn't told me. Also, still at Princeton, she, and also I mentioned that asking questions, you need to, one way it's an easy diagnosis to make, and in other ways it's difficult. You have to be curious and you have to ask lots of questions. Also, though, she began complaining about difficulty getting to sleep, anxiety, and we didn't know at the time that she had lost weight. I still understood this in my mind as being due to stress, and suggested that she see a counselor there, or that she go sign up for a yoga class and all sorts of things that she didn't want to do. She was more intent on taking her classes and coxing. And so she had these problems, but she also had an active social life. She had a long-term boyfriend and was very involved in what was going on at Princeton. For the rest of her years in college, let me just tell you the symptoms that surfaced. And you'll recognize those as some that Dr. Cunningham put up. And we have a brochure here that lists all the symptoms and talks to you a little bit more, or will tell you a little bit more about PANDAS and PANS. But she had sleep issues. She had social anxiety. And I'll give you one example of that. She came home and she had a photograph in a gallery exhibit that was opening up, which was the primary purpose that she was coming home. And she told me that she was just too nervous to go. She didn't know what was happening, but she just couldn't go. And there were a couple of other things that occurred when she was home that week when she said, I just couldn't go. She eventually did go, but she was clearly very nervous. Also home that time, she punched the door of her bedroom, totally out of character for her. Took her fist and punched it. We live in an old house, and I have a little bit more than that. And she punched the door. She had a binge drinking episode one night because she realized that alcohol would decrease her stress. We saw that she lost weight, she couldn't concentrate, and she developed checking behaviors and skin picking or excoriation disorder. Very episodic, and we thought it was related to the acne that she had new on her face. In her senior year, she did finally go to the health center. They diagnosed her as having OCD and generalized anxiety disorder. You can imagine what they gave her. They gave her Prozac, and she didn't take it right away, but eventually she did and found that it was minimally helpful. Now, I bring that up too because she's at Princeton in the health center. They had a record that she had been diagnosed with strep and with mono. I don't hold that against them. It was 2011, I think, when she went in there. 2012 when she went in there, and it just wasn't known. Plus, she was not a pediatric patient. Now, even Sue Sweeto acknowledges that those who are past puberty can indeed be diagnosed as having PANDAS and PANS. She went on after Princeton, and she got a job at Morgan Stanley. She'd had 2 internships there, but she got a job there, and her symptoms also increased quite a bit. But she continued to do great work, and I have a quote from one of her managing directors. He said, now, mind you, she has all these symptoms and she's having a really hard time sleeping. And if you know anything about finance people work from 9 to 5 in the morning, 9 in the morning till 5 in the morning. So it's really difficult to sleep. And it was hard to tell how much of her sleep issues were due to that or something else. But he said, Alex is the best banking analyst with whom I've worked. This is notable because she was not a finance major at, say, Wharton or Notre Dame. It was a technical skill, well, where you'd have a technical skill plug and play. But she was exceptional because, of course, she was smart, but she had a deep intellectual curiosity that drove her to ask questions and to go at things with energy and figure it out, whether it was in the healthcare domain or in technical financial skills, and she owned it. She was always very much invested in the outcome. So in spite of all the things that she had going on, and you see this in people with PANDAS and PANS, because it's so episodic, and in her case, she was at the mild end until the end. When New York was, in general, a great experience, but eventually she went to DC, and she made an appointment with Dr. Beth Latimer, finally. She would not seek treatment prior to that. And again, she said, Mom, it doesn't bother me that much. And by then she was an adult. I can't make her go. I can't involuntarily hospitalize her. But things were getting worse in DC. Now she had night terrors. She was sleepwalking and lots of other things. She became obsessed with looking at herself in the mirror on her cell phone. It was absolutely obsessed. And that was something new. And she also began to, what I would call, perseverate on certain topics, just absolutely not let them go. But she was doing well at work. She went to see Beth Latimer. She diagnosed her as having, and Beth Latimer, for those of you who don't know, is an internationally recognized neurologist and a real pioneer like Dr. Cunningham in this area of pandas and pans. And she diagnosed her as having pandas, and Alex was elated. She knew what was wrong with her. She prescribed a course of, or an infusion of rituximab and a course of antibiotics. But because she was doing so well, she went ahead and took a trip out west for her job. And glad she did. My husband and I met her in Tucson, and that was the last time that we had seen her. This was her last Christmas. This is her last birthday. I have never shown this picture before, but I'm showing it because this shows, she called, she told us she was sick, and that's a picture that she took that I found later on her phone of her when she was going into urgent care. And that's a picture taken 3 days before. So shortly after the picture on the right, on your right, she lost her life. And I will only say that this was completely, utterly unexpected. And she talked to me on the telephone. She hung up on me, which she doesn't normally do. And she took her Lululemon pants, tied them around her neck, and she hanged herself. Completely, utterly unexpected. So that gives you an idea why we founded the Alex Manful Fund, because we didn't know anything about this disorder, and we certainly didn't know that she could lose her life over it. So I'm going to rely on you reading the brochure there so that you know what we're all about, but it's basically increasing awareness and educating doctors. As I said, we so desperately need them, and research. And our focus is on adolescents and young adults, although certainly we work with children. We are proud of many things that we've done, including the Pond Brain Bank with Dr. Harris is going to talk about a little bit later, and that's where the term from the bedside to the bench and back to bed comes, because we thought by donating Alex's brain that we would begin to see some understanding of what's going on at a real microbiological level. He has done some research. He and his team there have done research, and I'll just say that they found mild gliosis and Alzheimer type 2 astrocytes, or metabolic glia, in the hippocampus and the basal ganglia. Based on immunostaining, there was a noted perivascular astrogliosis and an increased numbers of CD3, CD4, and CD25. So her brain was damaged. And that's what happens when people are not diagnosed appropriately, and or in the case of Alex, she diagnosed herself. I don't know if I ever told you, she had a lot of misdiagnoses. We have done some really exciting research, which I don't have time to tell you about now, but one of the researchers that we have supported, Lauren Breithaupt, who is now working with Dr. Jenny Frankovich, has found what looks like a biomarker. And they're so close, they are so confident of it that they have applied for a patent. I will tell you when we're outside that all the things that we're doing, fellowships and so on and so forth. I know I'm looking at an audience that can make a difference. You can't make up for Lulu. Many of you may know about Lulu. She's featured in her mother's articles, or a series of articles called The Hard Truths. Or Hudson, another young man who suddenly, and Lulu's the same thing, suddenly took their lives. Here's our mission, but our mission is there. We are, as an advocacy group, really try hard to get all sorts of information out there. And our local newspaper has been terrific, and the other newspapers. This is something I don't have time to go into. Our experience at NIMH was horrible, horrific. And we had to work with our Senator, Jean Shaheen, Maggie Hassan, to file for an investigation. But thankfully, and I feel it was meant to happen, that her brain went to Georgetown under the guidance of Dr. Brent Harris. This is the article that was published on her brain, and Dr. Harris, that's the piece of, the quote I read came from that, and he will tell you more. And well, there she is. That's from Tucson. I know I went way over, and I apologize for that. I was cutting, Nikki, believe me. And please ask me any questions that you might have later. Thank you very much.
So our next speaker is going to be Sean Miller from Yale, who's going to speak to us about SARS-CoV-2 and Alzheimer's disease.
Thank you so much, and thank you everybody for being here and listening, and thank you, Nikki. Thank you for the committee for having me come as well. So today, excuse me. So today I'll touch on some of the research I've been doing at Yale in association with SARS-CoV-2 and Alzheimer's disease like pathology in the central nervous system. And the title SARS-CoV-2 induced Alzheimer's disease associated pathophysiology in the human renal explant and organoid models, and how that all kind of makes sense of how these things kind of connect. So for today's discussion, I'll talk about AD or Alzheimer's disease, the antimicrobial responses of amyloid beta, association between SARS-CoV-2 and amyloid beta, and then I'll actually talk about a generation of a high throughput human model organoid system for drug screening against some of these compounds we find that mitigate some of this amyloidopathy, and then validations in ex vivo neurotissue from actual donors with Alzheimer's disease, as well as controls. And then lastly, the translation to the human long COVID clinic at Yale that I'm co-investigator of for the Yale Eye Center, where we're doing retinal imaging for proteinopathy. So Alzheimer's disease, I'm sure everyone in this room is very familiar with this. I just want to give one slide overview of prevalence, being that we have over 7 million Americans alone suffering from AD. Clinical symptoms include memory loss, cognitive impairment, and a lot of this is, well, I should say a lot of this is histopathologically recognized with amyloid beta plaques and then the entanglement of tau. Etiology, most cases are sporadic, really showing that it's, and with no cures, showing that this is pretty imperative that we find a way for solutions therapeutically. So amyloid beta, one of the histopathological hallmarks that I just said associated with AD. So amyloid beta, there's a hypothesis that it acts as antimicrobial peptide, or AMP, as it's an innate immune response against microbial pathogens. Previously shown in bacterial, fungal, and viral infections that the amyloids can actually encapsulate microbes to reduce spread and detrimental effects in the central nervous system, is which I'll of course focus on today. And you can see in this beautiful image is actually amyloid fibrils encapsulating bacteria. So SARS-CoV-2, so a virus that hasn't been necessarily associated with Alzheimer's disease just because of really the short-term symptoms have been really recognized. The long-term consequences of COVID-19, especially with patients who had severe or neuro-COVID or associated with such as brain fog and some of these other neurological impairments, has affected countless amounts of people. And so the short-term effects are known, but long-term not. And just to show you the kind of impact that it's had, there's 103 million confirmed cases in the United States alone as of September, with 750 million confirmed cases worldwide. So can SARS-CoV-2 actually infiltrate into the human central nervous system? Is this something that makes sense to even investigate? So in 2021, I apologize, there are citations there. In 2021, they had shown that Gomez had shown that essentially spike protein, I'm sorry, from SARS-CoV-2 could be found not only in the lung, but the choroid plexus, the lateral ventricles, and even in the cortex of human donors. So then my question kind of really came out is, does infiltrated SARS-CoV-2 induce an antimicrobial peptide response with amyloid beta in the human central nervous system? So in order to investigate this, multiple reasons. One reason of approaching this was I really wanted to find a translational model that we could actually really go to the clinic with. And I think there's a lot of talk, here in this group that we're trying to get everything we can to the clinic. So from here, I'm working with the co-director, Marcello D'Astasio, who's a neuropathologist at Yale. We decided to really try to see if we could do short interval post-mortem human ex vivo experimentation. So these donors donate their body to science, and then within a few hours, we actually have them at the morgue. And then we start, doing the experimentation that I'll get into. And really, this really preserves RNA and protein integrity rather than long post-mortem interval periods. And also, I'll show you that it allowed us to do some investigations with viable tissue. So before I get into that, I just want to kind of give my journey, the 8-minute journey to the morgue. So this is the building over there, past the subway is me. And then I run all the way past, usually sprint outside, so 8 minutes or less. And from that point is the second they take out human brain, so the whole brain or the eye globes, which I'll talk about, I'm running back to lab already. So, and then we're actually maintaining this artificial cerebral spinal fluid that was just bubbled, and then running it back and bubbling it for long-term culture, which I'll show you. So for this, we were just able to publish this in Science Advances of this year. We show that we can take these short postmortem, and I say short postmortem meaning less than 4 hours of PMI. They're harvested, we run them back, and then we actually, just to confirm that they were still viable, we do simple things like trypan blue, look at viability of cells. But what was really interested was, are they actually electrophysiologically active? Are these neurons still communicating in ex vivo tissue that was harvested from these rapid autopsies? And what we showed in the Science Advances was, yes, indeed, the human retina, the retinal neurons, we can keep them intact electrophysiologically. And then also we have a paper that just got rejected, but hopefully will get accepted somewhere else, using the cortex from Alzheimer's disease patients and culturing for these long-time culturing conditions for experiments. So then my transition is, so I'm mentioning, all this Alzheimer's disease, and we think of brain for that and the pathology associated with the brain. But one tool that we haven't really, that is underappreciated, I should say, is the human eye or the human retina, which is still an accessible part of the central nervous system. It's been shown that there are Alzheimer's disease-like pathologies such as amyloid beta aggregates in the retina of AD donors. There's a nice active neuropathological paper that did proteomic study on this prior to some of our examples in the Science Advances one. But what this is really about the human retina is that it consists of neurons, astrocytes, microglia, and a blood-retinal barrier, very similar to some of the architecture or some of the cell types you would expect in the brain. There's a lot of conserved pathological and physiological properties with the brain, and that's been shown through single-cell RNA sequencing, single nuke, and even spatial nowadays, and easily accessible with inexpensive and non-invasive clinical imaging. My background had been, before going back to academia, was actually looking at early disease detection. So my dream has always been, can we detect early so we can intervene earlier before the demise of neurons? So that's kind of where this rooted in me into the retina. And so what this is that, so these are the eye globes here to your left. And so that's what they look like when they come from one of our donors. And then I go through, and again, we're just doing this really quickly, is dissecting out. So then we're dissecting out the retina, which is a nice thing about the retina is it's pretty thin. So it's about 100 microns. So microscopy wise, you can actually get through there was epifluorescence over single photon or confocal. And it's very transparent, which is also very good for imaging modalities. So what we first did was we found donors, and so we found donors that had donated their body with age-matched controls who had no dementia, no cognitive impairment, and no retinopathy. So for example, sepsis is a rollout because sepsis is global inflammation. So we rolled those out, for example, and we found that there wasn't really any amyloid beta. There's some basal amounts here you can see in panel D in controls, just as expected, aging that you get some accumulations of amyloid beta. But you can see in the SARS-CoV-2 COVID-19 that there's increases of amyloid beta quantified in D. And then you can see that the co-immunofluorescence or co-localization between the green, the amyloid beta, and the red spike 1 protein, you can see in the COVID-19. Suggestive that if we maintain the integrity of RNA and protein, that we can still detect amyloid beta with spike 1 protein in the central nervous system. So then the question was, so we see spike 1 protein is co-localized with amyloid beta aggregates in the central nervous system. What is the receptor and what's the mechanism of action, the MOA? So what we did was this is just a really simple assay. We took just age-matched controls, human retinas that were from the rapid autopsy, and literally just bathed them with spike 1 protein, a recombinant spike 1 protein, and then applied thioflavin to visualize it. And we can see huge accumulations of the thioflavin increases, and you can see that with the GFP intensity and then in panel B quantified. Suggested that spike 1 protein was enough to exacerbate or induce some of these phenotypes that we see with amyloid. So next was, okay, well, what's the receptor? What is spike 1 protein actually working through? I know a lot of us in the room who are familiar with COVID and SARS-CoV-2 are aware of ACE2 being in the receptor. We found that actually from a paper that was initially published in Science of neuropilin-1 being a receptor for neurons to be infected with SARS-CoV-2 was indeed actually R-hit and not ACE2, and at least in the retina. And I say that because if you look, we did single cell RNA sequencing or single nuclear RNA sequencing, and we looked to see NRP1 and ACE2 expressions. And you can see that for NRP1 on the bottom x-axis, you can see that there's all the way up to 0.2, 0.3 is neuronal populations with expression of NRP1. But if you look at the same scales on ACE2, which is the y-axis, there's barely anything even 0.05. There's not much ACE2 expression whatsoever. So we went after neuropilin-1 as our target. So what we did here, yeah, the video's working. Okay, so what we did here was we took iPS cells, we took blood draws from human donors. We have controls, non-cognitive impaired, and we also have AD. In addition to, we have a Parkinson's story now too, but we actually then take those blood, we take the peripheral blood mononuclear cells, reprogram with Sendai virus at Yale, make them into iPSCs or induced pluripotent stem cells, maintaining that heterogeneity of our patients, make those into a retinal organoid. Remember, we're looking at the context of the retina. And then we've done things like spatial transcriptomics, proteomics, metabolomics, drug screens. But what do you really want to push on this is it bypasses the human tissue accessibility constraints. We get one donor maybe every 3 weeks. So our ability to drug screen rapidly is very much not feasible in that model. So what I wanted to do was have a human model that we could work on. So what we did was these bioreactors that you see in this video, each one of those little circles, and this is going at a consistent RPM, is a retinal organoid. These retinal organoids that are in there are months old. So they have photoreceptors, all the different mixed neuronal populations. And I batch them in bioreactors because there's hundreds of them. And so what I can do is do automated high throughput human retinal organoid drug screening. So these are 96 well plates. I have a very talented postgrad who can go and transfer individual organoids into these wells, optical well plates of 96 wells. And then we just screen them with compounds of interest with we first add in our fluorescence imager, which is crenad-28. We use it because unfixed tissue and it binds to amyloid beta 42. Fluoresces green very brightly. So we have our control condition, our experimental, which is basically we use for a positive control. You throw a lot of spike 1 protein on it. And then we screen different compounds and say what reduces that fluorescence intensity. It's very straightforward, but it's a very cool model. So we've been doing with epifluorescence for more of just rapid and global screening, but we've also done some single photon, which is something we're working on right now with high content imaging. So from working with this model, we actually then went through these NRP1 inhibitors and we tried a bunch of them. And what we ended up doing was we found this EG00229 NRP1 inhibitor that's commercially available. And we found that if you treat with tons of spike, of course, you get that increase in fluorescence with the crenad 28. But then this neuropilin 1 inhibitor, if you apply it at the exact same time as you're applying spike 1 protein, we see it mitigates a lot of this amyloidopathy or reduced amyloidopathy, suggestive that NRP1 could actually be that target that what we're trying to validate from our computational work. And then lastly, so we have NRP1, we have the kind of an idea of mechanistically what's happening. How can we actually get this information to the clinic then? So what we did was we found IRB that I'm co-investigator with ophthalmologist Brian Hafler at Yale. And for here is we actually have a long COVID clinic at Yale. And what we're doing is doing retinal imaging. So we just have you go to the eye doctor, they do that poof in the little eye and you get an image of the back of the retina. That's exactly what is happening here. So we did that. We have about 20 patients now that have already come through. We started this program back in February. So we're getting our numbers up higher. Hopefully we'll have more concrete results soon. But I wanted to show you that. So we're doing this imaging. This is real image from the Yale Center where you have a protein deposit. Again, we're not sure what proteinopathy it is. It's not labeled with anything, but we know there's abnormal protein accumulations in the retina of these patients with brain fog. That's one of our criteria for this long COVID clinic. And when we apply artificial intelligence, and this is machine learning tools that I've been developing in-house with one of my postgrads, and this is essentially this is our fundus autofluorescence in A, so just that image that you get. In B, it picks up everything that's hypopigmented. So anything that has like that white kind of signal to it. And this is just a really crude image of it. We have very specialized ones now that can pick up, you can do different parameters like sizes, of course, ferocity, but nonetheless is that we then take that, it automatically detects all the different dots or all your different stainings of interest or whatnot. For us, it's protein. And then C, you can see the overlaid. And then we quantify this. And it takes for about 10 fundus autofluorescent micrographs, so 10 people, it takes about 45 seconds. Spits out a number and it says, is it within our realm of saying like, do we think there could be an indication that there's something abnormal in the retina of that person? And one thing we're really working hard on is multidimensionality now in addition to this, where we're actually taking the clinical reports. So we're taking clinical reports in such as severity, such as other comorbidities as well, because no one's just a perfect clean, patient for our investigation, but we're taking all these things and seeing if we can make a better disease classifier at the end of the day, which essentially will be more precise because of all the modalities that we've inputted into our system. And so for this is, so we can do 10 within less than 45 seconds. It's user-friendly, unbiased, and it's consistent. So instead of someone biasly going through and looking at these proteinopathies or protein accumulations, the automated machine just, it just goes through it. And you can computationally get your results very quickly and consistently. And you can use your normal computer to do it. You don't need anything specialized, especially once we finish our code, you can literally just run it. And it's good to go. And then, so the future hope thing that is that we can do global, regular retinal imaging. You go to the eye doctor, you get your eye image, it goes through a software and it spits out what's your likelihood, or if there's anything abnormal. And then, so what does the future hold? So I think from looking at all of this together, I think that the high throughput human organoid drug screening there's a lot of opportunity there. Not only is it a human model, so it takes away a lot from cross-species compatibilities or incompatibilities, but it also, you're able to take and maintain that patient heterogeneity. So if we're talking about disease-specific or diagnostic or precision medicine, the ability to make high throughput, screen tons of organoids from that same donor could actually be a really good opportunity for us moving forward. And now with of course, with all the human donors that we get, huge, very grateful for them, is that we get to validate these targets in the ex vivo human tissues, which is universal. I mean, this is just, it's really cutting edge, I think. And then for this is that we're increasing our sample size too right now with the long COVID clinic for this pattern recognition, machine learning based models. And then the key takeaways being the Alzheimer's disease-related amyloid beta can act as antimicrobial peptide against SARS-CoV-2. Rapid autopsies from human postmortem donors can provide a very valuable tool to investigate disease pathophysiology. And then human retinal organoids can be produced in high throughput to screen drug candidates associated with viral-induced amyloidopathy. One thing I didn't mention today, I just don't have the time, is that with the high throughput, because when we give NRP1 inhibitor, it's the same time, but no one knows they're getting necessarily like, oh, I just got exposed to COVID or SARS-CoV-2, I need to take NRP1 inhibitor. So what we're doing is we have a compound right now that we're testing that is an anti-amyloid drug, but we find that it works very nicely at reducing amyloid levels after spike 1 protein in our organoid models. So we can actually treat with spike 1 protein, let it go for 48 hours, treat with our drug, let it go for 24 hours, and we see huge reductions in A beta. Suggested that it could be a post-infection treatment option in order to mitigate or reduce some of the excessive amyloid beta production. And then lastly, the human retinal imaging of neuro-COVID patients with the machine learning tools to really not only look for minor amyloidopathy, but also screen drug efficacy through the retina of individuals with AD. And lastly, and most importantly, is the acknowledgements. Dr. Brian Haffer, my mentor at Yale, and also the co-investigator for the Yale Long COVID Center, the Yale Retinal Imaging. He's an ophthalmologist by training. Marcel DeStasio, who is the co-investigator and co-director of the Rapid Autopsy Program at Yale, and then many of the team members who've been involved with the studies. Dr. Robert Logan, who's in the audience today from Endicott, who's worked on a lot of the computational work, and then collaborators for some of the compounds and also some of the images I showed was Martin Zhang or Dr. Kan Zhang at MGH and Harvard, Dr. Ran and Rudolph Tanzi as well. And then lastly, I just want to mention is that we also have Dr. Logan is going to present a poster later today, so hopefully everyone will see it. It's about Parkinson's and SARS-CoV-2 and looking at some of this co-pathology that we see. So thank you.
Sean, thank you so much. We really appreciate it. And now I'm welcoming Dr. Janice Bush from NC State. Just this to move forward.
Well, thank you all so much for being here. This is a real honor for me. I'm a PhD student, so I'm going to be presenting some of the PhD research from our lab at NC State. Nope. Or not. Maybe I can do it here. Oh, there we go. Oh, yeah. Do you need to? Okay, let me see if this works. Okay, perfect. All right, so I'm going to talk about microglia. And of course, microglia are the brain's innate immune cell. And they're responsible for not only mounting an immune response to incoming pathogens, but also to providing homeostasis to the brain, whether this is in terms of cell-to-cell communication or providing maybe neuronal growth factors and things along those lines. And microglia historically had been viewed as having sort of these two activation states where we are homeostatic, kind of copacetic, or we're inflamed. And this chronic inflammatory state is what is blamed for excessive pruning of neuronal synapses during Alzheimer's disease, which is what leads to the cognitive decline. So this is an image from a recent publication from MIT out of the Kellis lab, where they actually looked at single nuclear transcriptomics of microglia from brains with Alzheimer's disease across a really wide spectrum of symptoms as well. And you can see here that they have, I think my whole thing works a little bit. They had actually were able to identify in this paper 12 different transcriptomic states or phenotypes, if we want to think of them that way. Anything from some dysregulated homeostatic genes to inflammatory genes to the stress signature, a stress associated microglia. And we know that dysfunction for microglia is going to expand far beyond chronic inflammation. Microglia express a huge range of genes, not just the ones that we typically associate with inflammation, including most of the genes that are important to Alzheimer's pathology and risk genes as well. And I think also something that's really important to remember is microglia are actually known to be very long-lived cells, and estimates can go, I've read 5 years, I've read 20 years, I'm not sure about that one, but they live for a really long time. And so if we think about Alzheimer's disease being a pathologic process where clinical signs or the recognized signs might be happening 20 or 25 years after the disease is actually thought to start, it could be these glia are really impacting that brain health over decades. And I think another thing that's important about a long-lived cell when we think about pathogens is it might make them more uniquely potentially able to be infected for a longer period of time or just vulnerable potentially to the impacts of chronic infection. So our lab studies intracellular pathogens, primarily Bartonella henselae, and you might wonder why we picked this as a model to look at for Alzheimer's disease. So let me just talk a little bit about this. Bartonella henselae is a gram-negative bacteria, and you might be familiar with it from the term cat scratch disease, which is its common presentation. This is a self-limiting condition, usually children. And it occurs after the bacteria has been inoculated under the skin from a scratch or an animal bite, or even potentially from a flea bite from an infected flea. However, the spectrum of things that Bartonella can cause or is associated with is really expanding. And there are publications here on pandas from a few years ago. There's a recent publication on schizophrenia, and as well on mild cognitive impairment. So I think it is a pathogen that we should be thinking about in terms of some other conditions that it might be involved with. The other thing that's really kind of interesting about Bartonella is it really doesn't seem to like to play really well with other pathogens. And so there was a couple of papers, just speaking of SARS-CoV-2, that recently were published looking at recrudescent Bartonella infections following infection with SARS-CoV-2. And I think that that's something that might come into focus a little bit later in these multi-pathogen infections where maybe some of them are competing or just not wanting to tolerate one another being there. So for the project that I was working on, we used Bartonella henselae, the strain San Antonio 2, to infect the human microglial cell line, HMC3. And we basically then harvested those cells and we looked at the cell's transcriptional response at 6 or 48 hours following infection. Now, HMC3, if anybody's worked with that cell line, it's actually a fetal brain cell line, a fetal microglial cell line. And there was evidence in the literature that it would actually be more of a mature transcriptome if it were co-cultured with other cells. So I did co-culture it here with a neuronal cell line as well. They were co-cultured in these transwell systems. that did allow for chemical communication between the chambers. They were separated by 0.4 micron pores, so it did not allow for physical contact between those cells. And then briefly, just looking at the RNA sequencing pipeline, total RNA was extracted. We did ribosomal RNA exclusion. cDNA was then made that was sequenced on the Illumina HiSeq platform. So this is short read sequencing. And it was, I think, 150, or no, I'm sorry, 50 million. The depth was 50 million base paired sequences per sample mapped against the human genome. And then the DSeq2 package, statistical package, was used to determine differential expression in those infected microglia from their time-matched uninfected controls. And we did call statistical significance in this particular experiment as just anything that had an adjusted p-value of above 0.05 or below. That would be bad. So initial results showed us that these glia had thousands of dysregulated or differentially regulated genes, which I'll just shorten to DEGs, following infection at 6 and 48 hours, both up and down regulated. And initially, I tried to just kind of consolidate this data set to microglial genes that were shared in up or down regulation between both time sets. And you can see in the center there, that didn't really narrow my field down very much. So I then looked at genes that were specifically associated with Alzheimer's disease in some fashion. So either the genetic risk genes, genes that are involved in key cellular pathways, or I did go through a lot of literature looking at transcription levels, proteomics, and seeing whether or not there were some genes being targeted there. And in that case, then we came down to about 351 that were upregulated and 271 that were downregulated. Still a lot of genes, but much more manageable than, 40,000 or whatever it was at. So looking at overall what we saw when we started looking at these genes, there was this really wide range of different pathways or different components of the cell's life cycle that were being impacted. Everything, of course, from the immune response, which was expected, this was a microglia, to cell signaling changes, DNA damage repair changes, lots of things going on with proteostasis, protein processing, autophagy, and then mitochondrial function, as well as some interesting genes that might have been implicating some neuronal crosstalk might have actually been happening between that model cells. So starting with the upregulated DEGs, I tried to organize this heat map into kind of functional classifications here, and the colors there along the side are to match what I have here in the pie chart. But what I'd like to point out, a couple of things. One is that you can certainly see that these upregulated DEGs were primarily associated with cell signaling and the protein handling types of genes. The other thing that I'd like to point out is you can see here at the 6-hour time period, when we go between 6 and 48 hours, you sort of get this trend towards maybe a lower level of upregulation. So that's sort of that whiter color there on the color scheme. And that's something that might become important that I'll discuss a little bit more on the future slides. To highlight a few of these genes that I thought might be of interest to people. So these again are the upregulated DEGs. We had insulin degrading enzyme. This has been implicated in Alzheimer's disease. It actually may be somewhat protective because it also will break down amyloid beta protein. However, it may be playing a role in insulin resistance, which is something that's also seen in Alzheimer's. Potentially because there's competitive inhibition or just competition for this enzyme. If there's a lot of amyloid beta present, you're not going to have the insulin signaling that you might otherwise have or normal insulin signaling. IL-6, kind of a common pro-inflammatory cytokine, also upregulated. We know it's upregulated in the brains of people with Alzheimer's as well. P43 was sort of an interesting finding. We usually associate this with ALS or maybe frontotemporal dementia, but it is actually linked to Alzheimer's as well. And there were some interesting studies where they found it in inclusions in Alzheimer's brains. And then PCOM1, of course, is really known as an early onset Alzheimer's disease genetic risk. This transforming growth factor beta receptor was interesting to me because I'm always trying to think of what the pathogen might be trying to do here. So we know that it is implicated in Alzheimer's disease. However, when you look at brain transcriptome databases, you tend to see that levels are upregulated, which is interesting. You would think, well, that might actually be somewhat protective. In microglia, this gene is really essential for the microglia becoming homeostatic again, suppressing excessive activation. And when I think about my intracellular pathogen, it doesn't want excessive inflammation. So it would kind of behoove it potentially to drive the cell in that direction. PCOM, again, another Alzheimer's associated gene. We know that it is involved in phagocytic ability in the microglia. And we think that diminished function is potentially involved in Alzheimer's progression. And then lastly, the scaffolding protein, CD2 associated protein. There's genetic variants that are affiliated with Alzheimer's and that gene. Okay. So the downregulated genes, in much the same way, you did see that cell signaling and protein handling were kind of two of the big categories that were impacted, but also some of the immune genes started to really become downregulated here, and then saw quite a shift in metabolic associated genes as well. So that was a little bit interesting because, again, thinking of what might be beneficial for the pathogen, it's living inside of the cell. And going back to the shift between 6 and 48 hours of infection, again, in this heat map, you can see that it sort of goes from more downregulated to less downregulated. So it's kind of maybe coming back to the baseline a little bit. And I think, again, this is something with a pathogen, an intracellular pathogen, to keep in mind because it wants to hide. It's not wanting to announce itself to the immune system. And what we think might be happening here is that these cells are sort of slowly transitioning back to just their baseline status. Okay. A few genes that were differentially downregulated that are interesting. So ApoE, we were talking about this earlier, of course, a really well-known risk factor gene for Alzheimer's disease, typically upregulated in microglia across the Alzheimer's spectrum. But in this case, I wondered whether or not R. Bartonella could have caused this to be downregulated because of its lipopolysaccharide. It is a gram-negative. It doesn't have as stimulatory a lipopolysaccharide coating as other gram-negative bacteria do, but it's certainly something that we can look into. PTK2P is a drug or a gene rather that's thought to impact microglial chemotaxis. So suppressing this might actually prevent or slow these microglia from being able to go towards amyloid beta deposits or other things in the brain that they should be trying to eat. BIN1 is another Alzheimer's risk gene that we know that loss of function for this actually will impair the type 1 interferon response that microglia have. And then CDK5 has been implicated a lot for its function in Alzheimer's. It's dysregulated activity levels more than dysregulated transcript levels is my understanding when it comes to that gene. And then a few more genes here just to wrap it up. There's ABCA7, which we know is involved in lipid metabolism and processing and phagocytic clearance of amyloid beta. It's considered protective when you have higher levels of this in the brain, and it's downregulated here. And then downregulation of the PTPA protein. So it actually inhibits protein phosphatase 2A, which is involved in tau hyperphosphorylation. So if you're getting rid of your inhibitor, you might then be pushing yourself towards potentially having more phosphorylated tau. These next couple of genes here are involved with energy production, mitochondrial changes. And I think that's just another thing that's been publicized a lot in Alzheimer's is there seems to be this energy shift in mitochondrial damage. And then lastly, I put up this co-chaperone gene that's involved with proper protein folding. So another pathway that is also known to be dysregulated in Alzheimer's. Okay. So what does this all mean? I wish I had time to go through all the 40,000 genes. Let's stay for a few weeks. I think that at minimum, I think this research has shown that infecting our microglia with Bartonella-Henselae did have some significant impact on their gene expression and over this wide range of cellular function, many of which are dysregulated in Alzheimer's disease. So, I think about when I first started to sort of read into Alzheimer's, I'm a veterinarian, I've been a clinical vet for many years. Reading into Alzheimer's disease was a step in a completely different direction for me. So many different pathogens that seem to be implicated. And you start thinking, well, is there not going to be one smoking gun? I think there is going to be this multi-pathogen issue happening. What if you have a pathogen like this that's just creating these kind of slow, subtle shifts in a whole bunch of cellular realms over time? And that's actually then causing cumulative damage. So something to sort of think about and keep in mind there. Okay, so slow and subtle changes with Alzheimer's disease, I think, are the thing that we really see. This is a chronic condition, slow changes, subtle changes, persistent changes, things that really also describe our intracellular pathogens, be they our bacteria, our viruses, or fungi. So thank you very much for giving me this opportunity to speak. I definitely need to acknowledge my wonderful mentor, Dr. Ed Breitschwert, who runs the Intracellular Pathogen Research Lab at NC State, my committee members and our vector-borne disease diagnostic lab, as well as this wonderful Alzheimer's Pathobiome Initiative. Thank you guys so much. And also, I'm funded through the GAN Molecular Biology Training Program, forced me to get a minor in biotech during my degree. So thank you to them.
Janice, thank you so much. It's absolutely my pleasure to introduce Juan Fernandez from the University of Pittsburgh. And Juan, I apologize in advance. Everyone here might be verging on hangry. I don't know if anyone's hangry yet. Just kidding. We'll get to lunch a little bit later, but that's okay. We've had some phenomenal presentations. Thanks everyone for your flexibility with the schedule. And I will hand it over to you now, Juan.
Sorry, which one is yours? You can play the PowerPoint, the Fernandez. Oh. Okay, everyone, I promise I'll try to be as efficient as possible. I know everyone's probably got some hunger pains. So thank you for letting me speak today, and thank you for inviting me, Nikki. My name is Juan Fernandez. I am a recent University of Pittsburgh graduate, and now that's where I work as a research lab technician in the Orshamesh lab. I want to start by giving credit where credit is due. This is work done by Vanessa Hyde, which most of you have probably met, but she's a wonderful mentor to me, and she was nice enough to let me present some of her work. And I had the privilege of working with her on this. And this is some of our work that got published earlier this year. Sorry. So today I will be talking about a common virus that is seen throughout the world, and it's HSV1, and how it has implications in Alzheimer's disease. More specifically, how tau might have an interesting role that is often not thought about in Alzheimer's disease. Okay, so just a quick background. What we do at our lab is we study what we call infectious neuroscience, and this is a concept where we don't want to assign neurodegenerative diseases to just due to age or genetics. As Dr. Balin said earlier, genetics is only a small part of Alzheimer's cases. So the other 90-something% has to be due to something else, and we believe that infections are part of this. And so our group creates and uses cellular, molecular, nanoscale technologies to better image these pathogens and to better understand and treat these brain diseases. So I'm sure we're all familiar with the red flags of Alzheimer's disease, but I wanted to share some of the ones that personally interested us and we find motivating in a sense. And so one of them is that inflammation is in the brain. Obviously, Alzheimer's disease is described as an inflammatory disease. And this not only hints that the immune system is active in the brain, but it also, one of the biggest causes of inflammation is trauma or infection. And we also know that the disease is progressive in a semi-predictable manner. It has a frontal temporal distribution, which hints that the trigger propagates throughout the brain. And one of the things that we find particularly interesting, our lab, and I know my PI is highly invested in this, is that the fact that if you have a spouse that has had dementia, you're 6 times more likely to get dementia yourself. So this points at the fact of shared exposures. So these people are sharing beds, they're sharing cars, they're sharing clothes, whatever. And so we think there's something to look into there. And we also, someone mentioned before me, it's kind of hard to get AD, pure AD. Most AD patients come with pathologies, whether that's alpha-synuclein, which you see in Lewy body disease, or Parkinson's. And we also see TDP-43 pathology, which is linked to ALS. And so together, these red flags are something that motivated us to try to see if there's an infectious cause to Alzheimer's. And so this brings us to the pathogen hypothesis of Alzheimer's, which proposes that either viruses, bacteria, fungi, or prions have some sort of involvement in the development of these disorders. And so where do we start? One of the major suspects and more published viruses that's related to Alzheimer's disease, thanks to the work of Ruth Izaki, is herpes simplex virus 1, which has been found in postmortem samples and serological samples, though mostly through sequencing. And as useful as sequencing is, it does not give us a chance to explore the spatial context of this virus. So we want to see whether how it interacts with cell types and how it functions within the brain. So at this point, what do we know? Well, we know that HSV1 is found in elderly brains and also those brains that have Alzheimer's. And we also know that in lab models, whether that be in vitro or cell lines or brain organoids, we know that HSV infection causes AD-like pathologies. And we know that seropositivity in HSV is a big risk factor for AD. What we didn't know is whether we could find these proteins in human brains. We're big on the human model, and so we wanted to see if we could find these proteins because as people have found DNA, DNA could hint at infections from decades prior to death. So we wanted to see if we found proteins which are relatively short-lived compared to DNA, and that would hint that maybe there was a recent infection. So thanks to our collaborators, We were able to send some human brains, hippocampi to be precise. We sent it out for sequencing, and we essentially dug through a lot of data and found that there is, in fact, DNA. So we were glad that we found that too. And then one of our collaborators also did a huge mass spec analysis of common HSV proteins. Next, we wanted to further validate these findings by showing these in Western blots. And so we found HSV1 protein GB, HSV1 ICP27, and ICP4 as well in Alzheimer's brains. And well, the next logical step for us was like, can we image these brains, or can we image these pathogens in brains? And if anyone has tried to image brains, it gets kind of hard because viruses, one, are very small, so it makes them very difficult to image despite having top of the line microscopy technology or equipment. And there's also the fact that antibodies also have a size, and in dense tissue such as the brain, there's just not enough space to fully get a picture of what's going on. So a technique that we employ is expansion microscopy, which, to put it very simply, you bind down proteins of your biological specimen, and after a couple steps, you're allowed to pour water on it, and essentially it creates a hydrogel, which when you pour water on it, it expands up to 4 to 10 times its size. So this allows us to physically expand our biological sample. And so the concept is clear of what we wanted to do. We wanted to get some Alzheimer's brains, expand them, label, and image them. So an obvious question would be, is there any advantage to using expansion microscopy in aged human tissue? And the answer is yes, it's a big help. So one of the biggest factors is that the specific technique of expansion that we use that's more geared towards disease is the decrowding effect that the expansion has. So as I said before, when you pour water on it, it expands. And what this does is it physically opens up all these channels of epitopes that weren't exposed to you before. So you're allowed to see more of what's in there. So you can think of it as like upgrading your telescope. And second, this decrowding effect also eliminates lipofuscin, which is a huge factor whenever you're trying to image brains, specifically aged brains. So lipofuscin is a complex protein that just kind of builds up, and it's kind of just waste. And what this does is it absorbs light and it emits it right back at you. So you get false positives, and it also creates very noisy images. So it's kind of hard to see if you're to distinguish between your experimental labels and just false positives from lipofuscin. So we requested some brains from 2 different Alzheimer's disease research centers, the University of Pittsburgh and UC San Diego. And we requested for entorhinal cortex and hippocampus cortex, which are major players in Alzheimer's disease, and cerebellum too, because it has less pathology than the other 2. And we requested for this whole spectrum of the disease from no AD to mild AD and to advanced AD. And these were all age matched. And so we wanted to see whether we found HSV in brains. And not only did we see it, but we found that ICP27 expression increased with disease severity. So as the disease progresses from no AD to mild AD to advanced AD, you saw more ICP27, which is an immediate early protein. Not only did we see this increase in expression, but we also saw that the ICP27 shifts from neurons to microglia in advanced AD. So if you can see, we have the neurons and the microglia, and in no AD, there's It's highly correlated within the neurons, but as the disease progresses, it has more colocalization within microglia. So obviously next we wanted to see how it interacts with these pathologies in Alzheimer's disease. And so we labeled multiple tau phosphorylation sites and we found them. So it proved that we were able to use expansion to label p-tau sites in aged human brains. Next was to see what relationship this had with HSV1. And what we found was that not only are these both in there, but these two colocalize. And what we saw was that there's actually a gradual increase in p-tau burden as the disease severity progresses, which is expected. But we also saw that there was increased colocalization with p-tau and HSV1 proteins. So you can see here as it goes from no AD to advanced AD, we saw a gradual increase in colocalization. Next was to test the other hallmark pathology in Alzheimer's disease, which is amyloid beta. And what we saw was that ICP27 did not colocalize with either plaques or oligomers in the same way that it did with p-tau. Next, we wanted to replicate this in a model. So we used human iPSC-derived neurons, and we wanted to see if infecting with HSV1 led to tau phosphorylation. And this was done by one of our collaborators. And we, in fact, found that HSV1 infection in human iPSC-derived neurons leads to tau phosphorylation in a pretty significant manner. And we also labeled for total tau because we wanted to make sure that this wasn't just a general increase in tau, but rather a specific cause of the HSV1 infection. So next, after we decided to use brain organoids, which are a better biological mimic of the true architecture of the brain, and it actually showed the same thing. When we moved on to 3D brain organoids made by one of our collaborators, again, we saw that HSV infection caused tau phosphorylation and an increase in tau expression. This replicated the system closer to a real brain. Next was to see if we could do this in, just to be thorough, we wanted to see if we could do this in rodent cultures. And we confirmed the same effect in rodent cortical neurons. And HSV infection led to an increase in tau phosphorylation, matching what we saw in the human cells and the 3D brain organoids. Next, now that we had established that ICP27 led to this, we wanted to see if there was an inverse relationship between the two. And what we found, or the reverse, sorry, not inverse. We wanted to see what the reverse effect was. So tau phosphorylation on the expression on ICP27. And what we found is that once you use kinase inhibitors, we found that blocking tau phosphorylation increased ICP27 expression. So in other words, when tau wasn't phosphorylated, the virus replicated more freely and tau phosphorylation actually suppressed the viral proteins. And we saw the opposite with ocadaic acid, which just works essentially in the reverse way of what I just described. Next, sorry. We wanted to see, now that we had seen this, we wanted to see if human tau, specifically plasmid P301L, which is essentially just a easier version of tau that phosphorylates. If it can cause or prevent HSV1 proteins to express less or more. And what we found was that indeed when we used the human tau plasmid, we saw that indeed there was more expression. Now we wanted to test, now that we had found this, can human tau expression or phosphorylation relate to cell viability? And so we tested again, the same human p-tau, and we found that whenever it's HSV1 only, there's an 84% cell death rate. So only 16% of the cells are surviving, and that makes sense because you're stressing the cells. However, when we put wild type tau in, we found that there was a 26% death rate. So not only did the death rate decrease, but it was decreased significantly. And wild type tau is still able to be phosphorylized just at a slower rate. And then once we use this human tau, we found that there was only a 7% death rate, meaning that 97% of the cells were, in fact, or 93% were staying alive, which was a huge factor for us because it proved that the p-tau was actually helping cell viability. Next, we wanted to see a mechanism for this because we obviously it would have essentially helped us figure out more. And the route we decided to go down was the CGAS-STING pathway, which is pretty common in literature. It's a key component in innate immunity. And what we found was that CGAS-STING proteins co-localize with p-tau and HSV1 ICP27. So you can see here that NF-kappa beta, which are downstream effectors, NF-kappa beta and IRF3 are downstream effectors. They are abundant in AD, but also they co-localize with tau and HSV1. So this was hinting that the pattern that we found with tau and HSV1 also was directly linked in some way to the innate immune response. Next, we wanted to manipulate this pathway. And okay, you can see at the top, you can see that CGAMP activates the CGAS-STING pathway. So what we did was in neurons, we injected CGAMP to activate the pathway, and we saw an increase in tau phosphorylation and tau expression. You can see by the blue arrows. And then we did the opposite. We blocked TBK1, which is another downstream effector. And we found that the lights essentially turn off. So the inhibition of TBK1 prevents tau expression and phosphorylation. And just to summarize, we found that HSV1 proteins increase with Alzheimer's severity. We found that they shift from neurons to microglia throughout the disease. We found that ICP27 co-localizes with p-tau, but not with amyloid. And we also found that HSV infection actually drives tau phosphorylation in neurons and organoids in rodent neuronal cultures. Tau phosphorylation suppresses viral proteins and protects neurons, and that this all happens through the CGAS-STING TBK1 pathway, and it's mediated that way. Just quickly, I want to thank, obviously, this is a team effort. It was a huge collaborative endeavor. And again, I want to give a special shout out to Vanessa, who was the master behind all of this, and she allowed me to work with her. And I strongly encourage you, if I just rush through anything or you're curious about something, to shoot her an email. She's very nice. And lastly, obviously, none of this research is possible without money, sadly, and I want to thank our funders.
Juan, thank you so much. I wanted to mention that Juan is a member of our group called AlzPI Next Gen. We have a group of junior investigators with the intent of creating opportunities for them to engage with some of the more tenured folks on the team, as well as to, really bring together people that are earlier on in their career so that they can, work together with one another in our space. So thank you, Juan. I appreciate it. Thank you to all the presenters that went for this portion of the day. Lunch is ready. If I could just ask everyone to please return back a little bit before 1 o'clock so that we can sit down and get settled for the next, section of presenters in the afternoon. And lunch is out in the hallway. Thank you so much.